plenary session of Basic and Applied Sciences track of 15th International Research Conference of General Sir John Kotelawala Defence University. Thank you very much for joining us both in person and online today. We are about to begin the session under the theme of connecting science, technology, and innovation to achieve sustainable development for a secure nation. This session will consist of 5 renowned researchers and be co-chaired by Professor Charitha L. Goonasekara and Professor P.A. Nimal Punyasiri. To introduce and invite our co-chairs, I would like to call upon Dr. Sampath Biyalahakon, Coordinator, Basic and Applied Sciences, to the podium at this important occasion. Over to you, sir.
Thank you, Supun. Good afternoon, ladies and gentlemen. It is with great honor and pleasure that I introduce our first co-chairperson of the plenary session, Professor Charitha Goonasekara. Professor Goonasekara obtained her bachelor's degree in biochemistry and molecular biology with a first-class honors from University of Colombo, Sri Lanka. Then she earned her PhD in biochemistry from Memorial University of Newfoundland, Canada. It was followed by a tenure as a postdoctoral fellow at University of British Columbia, Canada. Currently, she is a professor in biochemistry, Faculty of Medicine, and Dean Research and Development at the General Sir John Kotelawala Defence University, Sri Lanka. She is the recipient of the KDU Research Excellence Awards 2020 for the significant contribution to research in the years of 2018, 2019 and 2020, and a recipient of National Science Foundation Research Awards 2018 for the Best Research of the Year in Biotechnology, awarded by the National Science Foundation of Sri Lanka. She has also been awarded presidential awards for the research published in the years 2003 and 2005. She has authored more than 45 journal articles and communications, and also she has 2 patents. Professor Goonasekara has held many prestigious fellowship, professional memberships such as Biophysical Society USA, American Association for the Advancement of Science USA, Allergy and Immunology Society of Sri Lanka, to name a few. Madam, I'm inviting you to the head table right now. Can you invite? Okay. Then, next, I would like to introduce our second co-chairperson of the plenary session today, Professor P.A. Nimal Punyasiri. Professor Punyasiri obtained his bachelor's degree in chemistry from the Institute of Chemistry, Ceylon, and earned his PhD in biochemistry from the Postgraduate Institute of Science, University of Peradeniya, Sri Lanka. Currently, he is a
recording in progress
at the plenary session of Basic and Applied Sciences track of 15th International Research Conference of General Sir John Kotelawala Defence University. Thank you very much for joining us both in person and online today. We are about to begin the session under the theme of connecting science, technology, and innovation to achieve sustainable development for a secure nation. This session will consist of 5 renowned researchers and be co-chaired by Professor Charitha L. Goonasekara and Professor P.A. Nimal Punyasiri. To introduce and invite our co-chairs, I would like to call upon Dr. Sampath Biyalahakon, Coordinator, Basic and Applied Sciences, to the podium at this important occasion. Over to you, sir.
Thank you, Supun. Good afternoon, ladies and gentlemen. It is with great honor and pleasure that I introduce our first co-chairperson of the plenary session, Professor Charitha Goonasekara. Professor Goonasekara obtained her bachelor's degree in biochemistry and molecular biology with a first-class honors from University of Colombo, Sri Lanka. Then she earned her PhD in biochemistry from Memorial University of Newfoundland, Canada. It was followed by a tenure as a postdoctoral fellow at University of British Columbia, Canada. Currently, she is a professor in biochemistry, Faculty of Medicine, and Dean Research and Development at the General Sir John Kotelawala Defence University, Sri Lanka. She is the recipient of the KDU Research Excellence Awards 2020 for the significant contribution to research in the years of 2018, 2019 and 2020, and a recipient of National Science Foundation Research Awards 2018 for the Best Research of the Year in Biotechnology, awarded by the National Science Foundation of Sri Lanka. She has also been awarded presidential awards for the research published in the years 2003 and 2005. She has authored more than 45 journal articles and communications, and also she has 2 patents. Professor Goonasekara has held many prestigious fellowship, professional memberships such as Biophysical Society USA, American Association for the Advancement of Science USA, Allergy and Immunology Society of Sri Lanka, to name a few. Madam, I'm inviting you to the head table right now. Can you invite? Okay. Then, next, I would like to introduce our second co-chairperson of the plenary session today, Professor P.A. Nimal Punyasiri. Professor Punyasiri obtained his bachelor's degree in chemistry from the Institute of Chemistry, Ceylon, and earned his PhD in biochemistry from the Postgraduate Institute of Science, University of Peradeniya, Sri Lanka. Currently, he is a
Recording
professor
in progress.
and chair of
and also to have him introduced at this session because he has been one of my teachers as well. So Professor Nalin Silva is the chair, senior professor of chemistry at the University of Colombo. He obtained his BSc from the University of Colombo and PhD from the University of Cambridge, UK. He has gained postdoctoral experience from University of Cambridge and Louisiana State University, USA. He worked as the science team leader in Sri Lanka Institute of Nanotechnology, SLINTech, from 2012 to 2019. Upon completion of the assignment at SLINTech, he returned to his university position in 2017. He has published over 100 SCI indexed papers and 4 US patents. His present H index is 31. He has won the Presidential Research Award for 10 consecutive years and a National Research Award presented by the NRC and the NSF, Sri Lanka. He was also named the Young Scientist of the Year in 2004 by the World Academy of Science and the NSF. And also he received the Vice Chancellor's Award for Research Excellence in 2017 and 20 from the University of Colombo. He was a member of the National Nanotechnology Committee and chairman of the National Nanotechnology Research Panel at NSF. He was also a member of the committee at NSF for drafting the National Nanotechnology Policy of Sri Lanka. He also served as the chairman of National Basic Sciences Research Committee and National Committee for Technology at the NSF. So his main research focus areas are advanced material for healthcare, water purification, textiles and apparel, natural resources and nanobiotechnology. So recently he was appointed as the chair of chemistry and also as the head of the Department of Chemistry, University of Colombo. Recently he was awarded with the most prestigious professional qualification, that is the Fellow of the Royal Society of Chemistry from United Kingdom. So we would be hearing a glimpse of what he has been doing throughout his career. Sir, it's over to you, sir.
Thank you very much, Charitha, for your kind introduction. And I'm very happy to be here. And also my collaborator, Professor Nimal Punyasiri, also here. Thank you very much, both of you. And especially Charitha was one of my students in the Colombo University. I'm very happy to see that she's a dean research and how she progressed to this level. And to say a little bit on the KDU, I have experience in teaching here also, allied health science and the Faculty of Engineering also. I have taught many courses. And I don't know whether right to say this, I really like the discipline. I have been telling Nimal also how disciplined the students were during lectures, and it's not the artificial thing, it's a real, that is where I strongly believe that the discipline takes you to the top. So, and also I just talked to him, he's also one of our students. There's another colleague here. And the structure of KDU, I think we must take that as a model to the Sri Lankan education system. We are struggling in the Colombo University. We have the best brains, but there are the funding issues. This is a real model. That's a compliment for the KDU. I have told this in many places. I mean, I openly tell without any political interpretations. Okay, I have few, another important factor was the discipline. You started really on time at 2 o'clock, 2:10, you handed over the podium to me. I'll hand over to you 2:30. So please ring the bell after 15 minutes because Sagarika also worried that I have 60 slides to tell. Yeah. Okay, thank you, Sagarika. Yeah. So my topic is electrospun nanomaterials for drug delivery, tissue engineering and cancer therapy. My background was physical chemistry. My background was mathematics, but I had a real fascination on the biology side. And my collaborator, the co-worker, my wife, Professor Rohini, and she's on more inorganic side. And so we build a nice team in the Colombo University with adding more collaborators. So this, we have also, like Charitha mentioned about my CV, a brief bio one, we have water purification, other advanced material research, but biomedical research, we think we can do something for this country. This is really where you can really impact on the society, because the science should be, ultimately, it should transform into the society. I will take up the theme also, because I think I will take 5 minutes, more than 5 minutes probably to discuss this before I even come to my topic, because these are very important things. All are now the Sri Lankans here, there are something people joining through Zoom, but we have something to change our mindset to how we take the science forward. That's very important. Conferences are really beautiful, but how do we transform that into nothing wrong with the universities or the academic staff or the researchers, but there are some things that we have to address openly, and the people will take the message on that, because we are the people who should transform that message to the society. I think this is a good forum to tell a few things about that also. All right, okay, it's not moving. It's not moving. Enter. No. Clock stops. Like the football games. Right. They got okay, right. That's fine. Fantastic. Better than that. Technology, I was given the chance to come through Zoom, but I really don't like Zoom. Now we have to be normal. That's why I'm not wearing the mask also. No, everybody must be thinking why I'm not wearing the mask, but we have to come to the normal. Okay, right, fine. So okay, I'll try to quickly go through this because there's an international conference. I thought some sharing this beautiful country. Everybody is now, one of my, brothers was planning to take the passport. He said 3 months you can't get the passport because the people are flooding to go. But remember that grass is green elsewhere. You think that the grass is green? No, grass is same in everywhere, right? So think about that. If you try to go, I just want to show you some nice pictures to motivate for you to do something for this country when you go back and come back and serve this country. Because this is a beautiful country. We have to develop this country. Right, 17th century, this was a lifestyle 17th century. And You know, Einstein once said that we will get back to 17th century. Now we have been now experiencing the fuel crisis. You get back to the firewood, cooking, all these things. So, but after 17th century, you know, the industrial revolution came after the world wars and so on. And second industrial revolution came, and the third industrial revolution, IT and so on. And then you are now with the next generation revolution. The pace at which we call this is a exponential or the destructive innovations. One innovation takes off 10 products from the market. Like your mobile phone took off about 10, like the radio, TV, the voice recorder, video recorder, all were taken out from the mobile phone, one device. That's a danger of this society. That's what we as scientists, you know, we have to be in par and in pace with the outside world. Otherwise, we will be importing the technology every year. We have been a developing country for the last 50 years, and still we are a developing country. Now we are a bankrupt country. So it has now even gone worst after 50 to 60 years time. So where is the beginning for us? We have the big brains, I tell you, right? But the danger is the world is moving. Economic recessions are everywhere. The Americans feel it. Europe feels it because of the Ukrainian war. And the Taiwan and all these countries, they feel that stress. But at the same time, the science and technology program goes on. That's the beauty of so therefore, you have to think about, this is how the spacecraft looked like in 1961. So many, the buttons and devices. 1981 to 2, space shuttle came into picture. Now you can see Dragon 2 in 2019. It's only a few panels, all touch screens. The technology. Elon Musk is the richest man in the world, created this. With one person's vision, you can see how the technology advances. You know, the space suit in 1960s Apollo mission, look at the size of the people. Now look at the Crew Dragon, the size. Because what's that? This is advanced materials. It's a technology. That's, you know, the materials. Now that is to prevent them from high pressures, high temperatures and so on in the space. But look at now, look at the technological advances. They are like normal human beings without any other things. But all features are there in that. So this is the technology that we are talking about. Now you look at the Elon Musk future generation car in the Tesla, you know, you don't need to drive even, right? I will not do that because I like driving. I like fast driving, right? So, but anyway, you can get a taxi to your home in the future. You don't have to worry about. So the drivers, the jobs are going to be taken over by the machines. Now, the next thing is now I don't know how many you follow the technology. Day before yesterday, they completed the DART mission. They hit an asteroid head on with a spacecraft. You know, these are the technologies that we are talking about in the world. So now where are we at the moment? Now, these are the 5 technologies that will shape up the planet in next 20 years. Nanotechnology, biotechnology, artificial intelligence, robotics, and IoTs, Internet of Things. You know, if we are not geared our science community into these technologies, you know, we are going to be redundant in another 20 years time. Our people will be jobless. Now, if you look at the second richest man in the world, the Jeff Bezos, he is the Amazon owner. If you go to a shop in the US, he has shops. No one works there. Fully automated, artificial intelligence, robotics. You can see. So this is the danger. What we do is science, that we are doing science. But where do we stand after 20 years? We'll be same. What I think is how the things are moving. But the individually, institution-wise, individually, we may have things for us to boast. But country-wise, we are in a really dangerous situation. So the theme of today's connecting science, technology, and innovation to achieve sustainable development for a secured nation. It's a fantastic theme. Now the achieving is, I talked about Sri Lanka crisis. I have no time. I have only 10 to 15 minutes to talk about. I would love to talk maybe longer time on addressing all United Nations Sustainable Development Goals, because this says sustainable development. Then you have to achieve the SDG goals. If you look at one by one, these are the 16. The most, I have no time to discuss these things, but the number 16, it's the last, but that's That's where we should start. Peace, justice, and strong institutions. That is where we must start. If you start that, all things will work. But it's the last thing that we will probably see in this country. But that's beyond our control. So these are the 7 problems that we will see as scientists in the next 20 years that we will see. I usually, I took this slide from my wife's presentation, so she must be looking at it. And a little bit change. Normally we put the cures for diseases such as cancer and COVID-19. Normally we usually put it at the last, but last 2.5 years you saw after the pandemic and so on that came into the top. So we will be struggling. You talk about the COVID-19, but you will get more things in the future, you know. So the more people deviate from the nature, the more the nature will punish us. Okay, that's a real equation that comes from your religion also, what you believe. Cheap and clean energy. We have already encountered the problem with the cheap and clean energy. So we are really struggling without energy. I have no time to discuss how the solar energy can be harvested in this country, how we can be self-sufficient in energy. We have the best solar scientists in this country, but no one will be benefited from any of the policies that we have on the energy. We always depend on the fossil fuels and the other alternative fuels, but those are not sustainable. Look at now, we are experiencing, we are discussing that another 10-hour, 12-hour power cut come because of the coal. Why not sun? 12 hours, 10 hours, we have sunshine, but we are not harvesting. Increased demand for pure water. We have a crisis in the North Central province due to the CKDU. So we are doing work on that also. Reduce environment pollution, computing power, world hunger, defense research, and so on. So those are the problems that we will encounter in the future. Right, this is my main topic, the electrospinning. We use a simple technology. We work with the nanomaterials, nanotechnology, creating small, I'm not going to, discuss what is nanotechnology. Nanotechnology, simply it's a manipulation of the matter at the atomic and the molecular level and making useful functional materials, manipulating the matter at that length scale. That's a nanometer, 1 to 100 nanometer length scale. So we are working on some fibers and using the technology electrospinning. And this is not a new technique. This has been there for 100 years. Okay. So the technology wise, it's an old technology. People use it, this electrospinning, but we use, especially for right-hand side lower corner, you can see the wound dressings, drug delivery, and tissue engineering scaffolds. That's where we are targeting electrospinning, especially drug delivery. And if you look at the electrospinning basic technique, anyone can set up. We have two set up, two apparatus at the Colombo University. You need a high voltage power supply, DC power supply, and a syringe pump, and a small perspex box. You can buy it even from the plastic vendor, supermarket, or somewhere. You just apply a high voltage, and the liquid pushed through the needle. When the electrostatic force overcomes surface tension, then it will eject as a droplet. You can get it, or you can get it as a fiber. It falls into a. I can't play the video. So you can basically get like these fibers, right? It's not. It's easy to set there and done, but when the person who is doing it, Chaturika did it. Chaturika, you did, right? Yeah, Chaturika was one of our students. So you can create nano level fibers, you can create micro level fibers, you can change the applied electric field strength, flow rates, properties of the solution like the concentration, viscosity, conductivity, surface tension. You can change distance between the syringe and the collector. You can manipulate that, and ambient parameters like the humidity, temperature, and so on. All these will affect the fiber. So it's not very easy to do it, but a competent person can get a good quality fiber. And then we can have the fiber in one polymer solution. You use the polymer melt we call. You can use a one polymer or two polymers. It can be monoaxial or the coaxial or side by side or triaxial. So that means if you look at the end-on weave, you can have one polymer or you can have two polymer, like the core and the shell you can have, and you can have side by side two polymers. It's like attached two strings, and then the other one you can have three. All these layers you can put whatever you want into the fiber. Mix it with the polymer, you spin it, you will get that material. So what we do is we have the drug delivery application, especially with that one. Normally, if you want to coat a drug on a fiber, the loading is very little, but what we do is we melt the drug with the polymer, and then you spin, you get a loading capacity is high and high encapsulation efficiency, all delivery systems and so on. And these are some examples that you can see it. And coaxial electrospinning is not very easy. It's difficult because you are having two polymer blends and you have one inner, one outer. Outer you can get, if it is a drug delivery, you can get a quick release drug, and the other one you can have a longer release drug. So that means you can tune the drug delivery according to your wish. Triaxial, these are some examples that you can see. And then these are the drug delivery profiles that you can have. The holy grail of drug delivery is a zeroth order, that's a green line. Everyone prefers a drug to be delivered continuously over the period of time. But some are fast dissolving, like if you have a headache, you don't want a drug to be delivered after 10 hours. So you need a fast dissolving drug. Like that, intermediate, immediate, delayed, extended, pulsatile. All these things can be manipulated using the electrospinning technology. So these are some examples. Antibiotics, antibacterial, all these things can be incorporated into the fiber. So smart textiles also will come into picture. The textiles will come like, with the antibiotics or the anti-cancer drugs, maybe the undergarments of ladies because of the breast cancers and so on. And you can create the undergarments, slow releasing anti-cancer drugs inside the electrospun nanofibers and use that material to knit these kind of undergarments. So those things are all, you can make IoTs, and you can make garments communicate with the outside world through a device where the doctors get all the information about our body. So those are all possibilities. These are some examples of drug release profiles, some of the publications that we have. Again, some you can see the left-hand side, you can see different release, cumulative drug release percentages given in, because you change the polyvinyl pyrrolidine to ethyl cellulose polymer ratio, you will get a different release profiles. So that's the beauty of that. So whatever the release that you want, depending on the disease, you can get the real the drug release profile. Okay, some of the publications. So this is an example that you get a almost zeroth order drug release for all compositions, right? So this is one of the work that we did it during our Slentech time. This was a high impact journal, Applied Materials and Interfaces, American Chemical Society. There we embedded antibiotics on a halosite nanotubes, again electrospun. So these are some prototypes we have made and we have kept in the laboratory. No one comes to commercialize, so we keep it in the laboratory, we publish, and we are happy. All is well, funding organizations are happy, we get money, we publish, and our H-index goes up. So the country is going in the same pace, right? So these are some things that if somebody wants to take it forward, and we have these wound dressings, our PhD student, Ms. Ume Angana, is expert on making these scaffolds. You can make it inorganic nanoparticles, you can make metallic nanoparticles, silver, gold, you can incorporate there, and even the zinc oxide, titanium dioxide, anything you can incorporate into the fiber, right? And also we are now trying to do the Ayurvedic Rasa Shastra also. Ayurveda uses mercuric sulfide as an agent, that we can use that also, because we have to believe that the indigenous medicine has a role also to play. So we don't discard indigenous side also. We got a PhD student and she got a graduated also with this technology, extrapolating into the Ayurvedic system as well. Skin tissue generation, final few slides, Charitha. I think the time is over now, right? Yeah. Okay. Give me 1 minute. Skin tissue generation, we can do in the future. Artificial organs, artificial blood vessels. Those things are going to come into picture. This we can do it using the electrospinning technology. The people have done outside the world. Artificial organs, you can make it. Electrospinning. This is artificial blood vessel. You can do it using the electrospinning. What you do is a collector is a rotating disc. You get the vessel. You can do it very easily. Bone scaffolds, anti-cancer drugs, you can do it. And cosmetic face mask, we can do the nano gold. Our Dr. Danushi has done this work. It's very effective cosmetic face mask using the electrospinning technology. Water purification we have done, and we can commercialize this. This is the industrial scale. We don't have money. If somebody is interested in, we can make it ton scale now, this material. So that's all. These are the collaborations we have. University of College London collaboration. That's our School of Pharmacy, and they donated us some money through the EPSRC scholarship. And this is the electrospinning material. Recently, we had a book chapter on electrospun nanofiber, especially electrospun fiber dragna. So this is our lab. We have a Center for Advanced Materials and Devices lab. We work with Professor Mandika Vijayaratna for wound resin applications because we need the clinicians to come into the picture. That is our PhD student. That is Professor Rohini sitting in the background. And these are our PhD students. And it's slow, sorry. These are our PhD students, a lot of graduated. And you can see all of them are now working in this is moving slowly, Charitha. Oh, yeah. These are all now graduated and working in universities as lecturers and, the private sector and so on. So the presentation is disappeared. That means end of the story. Yeah. Thank you very much for listening. I'm happy to answer any questions that you have. Yeah. Thank you again.
Thank you very much, sir, for very interesting speech, and also for keeping on time. Right. So the forum is open for any questions from the audience.
That's what normally happens. University College London, we work together, and they might commercialize, but these are not. We have not gone through the patenting also. Patenting also very expensive in this country. How can you make a patent? We are struggling to make a patent also. What we do is we publish. When you publish the work, it's in the public domain. Right? Anybody can do it. So these are the technologies that's expensive for us, but I'm talking about this to mass scale production. It's not a difficult discussing this with Aston Pharmaceuticals also, but the problem is them also the bandwidth and to launch a product or a commercial aspect, it's very difficult. But the Korean model, if you look at it, Korea, the last 40 years, the first 25 years, the government did this commercialization. Then they slowly handed over to the private sector. But now we are asking the private sector to come, but they don't, we have to discuss about their product portfolios, and they are bothered about the return of investment. If the return of investment is 20 years, they will not invest. They are the low hanging fruits they want. But the longer return of investment, the government should put the hand onto them. But it's not happening. No one understands this equation. But we tell this forum, but I hope somebody will be there to take this message forward and tell how this can be commercialized. Good question. I mean, can do it, yeah. But they will do it. They will do it, yeah. Yes. The reason is they don't understand what is commercialization means. They think the lab level experiment can be commercialized overnight. There is something called the innovation death valley. You have to go from the lab level, 1 gram level to 1 ton level. There is a huge bridge you have to cross. That can be broken at some point from prototyping to large scale to even the commercialized. Some products in Korea, they failed at the market level. After launching, the people didn't like it, it fell down. That's that shock. The private sector doesn't want to take that risk. You know, somebody must take the risk to do it, because they think that when the basic scientist develops something, it can be commercialized. It's all nonsense, because they don't understand, and there are no people to make them understand as advisors. I'm telling you point blank. That's the problem here. So it's not easy to commercialize. Normally commercialize a product from a lab level prototyping to the product 2 to 3 years minimum. Some products 10 years. You know, a lot of troubleshooting to be done, because before you go to the ton scale. Yeah. So I mean, somebody must start a discussion on this. We must discuss openly where we want to go. But I'm fully against for blaming scientists. Because scientists are because the reason is these scientists go abroad and flourish. What is the reason? Can't we analyze that reason? You know, they go to the NASA and we have 200 Sri Lankans working in NASA. Maryland, if you look at the engineering school, we have a lot of scientists. Every MIT, every Harvard Medical School, all Sri Lankans are flourishing. Why not here? We are flourishing, but we are not visible. That's the thing. So visibility comes with the companies. Now you can see, even at least I wanted to start up a company with my innovation. We have no laws in this country. An academic staff member cannot start a company and become a working director. How can we develop this country? MIT Rob Langer has 100 companies. He's a founder director for 100 companies in MIT. Robert Langer, you go and see his index is 189. Still, he holds 100 company shares. Where are we going? So that's the situation. Yeah. So don't, I mean, I'm fully against blaming our scientists. Our scientists are the best in the world, continue to be the best in the world. Yeah. Hey, Kalpa is here. You see, all our products, no? Huh?
Yeah, I think well said, sir. I think it's, yeah, the duty of the scientist and the academics is something, and all the innovations and everything comes with great investment as well. Taking risk also is a part of it. So I think it's the government or somebody who has the authority, its role to step in and build that bridge. So yeah, we hope for the best for the future. Yeah, are there any questions? I think the time is also closing up. So because of that, we'll advance to the next speech. Thank you very much, sir, for the very inspiring
Thank you very
words.
much.
Let's move to the next speaker. I would like to introduce our second speaker of the session, Dr. Andrés Fernando Muro. Dr. Andrés Fernando Muro is a group leader of Molecular Genetics Laboratory of the International Center for Genetic Engineering and Biotechnology, Italy. He obtained his PhD in biology from University of Buenos Aires, Argentina. Dr. Muro's main focus of research is the understanding of gene function and their relation to diseases, and the development of efficient therapeutic approaches using mouse model with special emphasis in metabolic liver diseases, especially in the study of mechanism of diseases and possible therapies of neonatal hyperlipidemia and Crigler-Najjar syndrome type 1. That's very important, inborn errors of metabolism. He is interested in understanding of mechanism of bilirubin neurotoxicity in order to develop pharmacological and gene therapy approaches aimed to avoid severe and permanent brain damage and lethality. Very important area of research with the current health aspects. Ladies and gentlemen, please welcome Dr. Andrés Fernando Muro.
thanks a lot for your kind invitation, especially for the KDU and for to Ihani Hayaguira for the invitation to this interesting meeting. So, and thanks for the introduction. So I will try to continue with this inspiring talk that we have just listened before, and I will try also to tell you some other technology that we are since the beginning, since already 50 years, trying to transfer to the, let's say, to the public, to the patients, and I think that this is the right moment to say that this is really working and to show you the potentialities of that. I will start sharing my screen. Okay. Okay, can you see my screen? Yeah, you see that is there on the board. So I will tell you today about the potentialities of gene therapy to treat human genetic diseases. So this is a very interesting situation because about 50 years ago, so at the moment in which genes were still not known, which was the structure, DNA was not possible to sequence, it was just a few years after the restriction enzymes that are the tools that allow us to do genetic engineering, really. These were discovered in the late 60s, and I am talking here about this paper in the early 70s, in which it was proposed to use gene therapy to cure genetic diseases. And this is really it was a pioneer work from Friedman and Roblin, in which so they hypothesized a system in which a metabolic pathway, let's say, could be altered. In this case, for example, the enzyme number C. So sorry, the product number C is not being produced, or is being produced alternatively to a compound number X because of a mutation in the gene number 3 of this putative metabolic pathway that results in the mutation of enzyme number 3. And at that moment, as I mentioned, very poor knowledge of the biology was available, but some groups already started to introduce with viruses into cells, into tissue culture cells, just the missing function by and also with exogenous DNA or naked DNA. So these were really times in which very few information was known, and instead, well, they were able also just to establish some preliminary criteria that are still today used. Think that this was done 50 years ago in 1972. So the idea to have a very good characterization of the genetic disorder, so to know experience, to know if other treatments could work in order to compare with our new treatment, obviously to characterize the DNA vector, but they still didn't know how to make it or how was the composition, and to use animal models to study benefits and adverse side effects. And obviously the only tool they had at that moment was to test this in patient fibroblasts. So these are really, let's say, the starting points of what we are doing also today in gene therapy. And over years, gene therapy evolved, and they started to use a different systems. In this case, the first system that were used were just retroviruses, just to treat immunological disorder, and the main one that was used at that time, we are talking here about the late 80s and early 90s, it was an immunodeficiency that has a very complicated name that is called severe combined immunodeficiency due to adenosine deaminase deficiency. And these are, you may have seen these kids early on. Now they are not so frequent. So these are the cases that do not have any immunological system, and they have to be inside a plastic bubble in order to be isolated from any kind of pathogen that could be very dangerous for their life, because they don't have any immune system working. So these were the first clinical trials ever done, and in most of the cases they work using this retrovirus, but unfortunately, some of these developed some tumors because these retrovirus were inserted into some or nearby some proto-oncogenes. And this really happened, and this was a very negative situation, not only for the patient because they had the tumor, but also for the field. Also in this period, in the 90s, another very, let's say, negative situation appeared, that in a clinical trial for an urea cycle disease, that is ornithine transcarbamylase deficiency, there was a fatal result. As a patient that was treated with adenovirus gene therapy died. And as I mentioned before, this brought the field of gene therapy really from what it was at the beginning, that was a flourishing field with a lot of expectatives. It went down to a position to start to work very hard to improve the system and to give more safety and efficacy to the patients. So new vectors have been developed in the years. The ones that we are using now are always also viral vectors. One of these is adeno-associated virus that has very specific characteristics. It is not pathogenic. Another one is lentivirus, that are vectors derived from actually the HIV virus, that were modified in such a manner that in both cases to limit as much as possible any pathological consequence. We are also using now other technologies like lipid nanoparticles, in which we can encapsulate mRNA into the, or let's say nucleic acid inside the particles, and also virus-like particles in the same manner. So these systems really work, and we have now in the market different products. The first one was already in the commerce, approved by the EMA, the European Medical Agency, in 2012, and then we have other products that have been approved by the MAA or the FDA, the European or the US agencies, for treating of different rare diseases like spinal muscular atrophy or some particular kind of blindness. And why we're interested in rare diseases? Rare diseases, as such, the name itself is indicative of that they are rare. But if you put all the rare diseases together, let's say, from which more or less we have more than 1,600, about 70% are of genetic origin. And if we put all of them together, more or less 3.56% of the population is the prevalence of rare diseases in the population, with correspond more or less to 260, 450 million people affected globally. So this indicates that it's a lot of people. We are talking about a lot of people that need really a cure, and normally for these rare diseases there is no cure. So as I mentioned before in one of the first slides, we have here our metabolic pathway, in which we have a mutation in some of the enzyme, and then we have deficit of the product. We may have also accumulation of a toxic substrate, and then activation of other secondary pathway that may not be as efficient as the original one. And this inborn error of metabolism, the liver is the main participant because of the high number of events that occur in the liver. So in that case, we are involved, the main organ we are studying is the liver. And for that, we use adeno-associated virus just to deliver our DNA into the liver. So this virus that contains specific genes, viral genes, are removed. And when I remove, we insert here our therapeutic gene under a tissue-specific promoter. And this makes that the virus, when it enters the hepatocyte, it remains as a circular episomal chromosomal instead of being integrated because of the absence of the enzyme. We can also use different virus to target different tissues, that this is called tropism of the virus. So in that case, what we are doing is we are using our therapeutic product, that is our virus, and then we in vivo infuse this virus into the circulation, and it reaches the liver, and we use this for different diseases such as hemophilia or some eye disorder, or urea cycle, as I mentioned before, or Crigler-Najjar syndrome, that is our topic today. This has been proved to be very efficient in hemophilia. So you can see here that the patient that were originally with no factor 9 activity, these are hemophilia B patients. After receiving the infusion of the virus, they have a very good activity over time, and they did not receive any more infusion of their recombinant factor that normally they receive once every 1 or 2 weeks. So this makes really a change in the life of the patient. And in the case of bilirubin, I will show you our latest result, in which bilirubin, as you may know, is the degradation product of the heme molecules that are derived mainly from the red blood cells. And these are converted into, the heme is converted into unconjugated bilirubin by bilirubin reductase. And this unconjugated bilirubin normally is conjugated, is insoluble molecule and needs to be eliminated in the bile. If not, it accumulates in the lipid-rich tissues. And when this enzyme, UGT1A1, is not present, what it happens is that it goes into lipid-rich tissues as the brain accumulates there and produce damage and produce permanent brain damage and death. When the mutation, we have the mutation in the gene that is very rare, that it affects more or less 0.6 to 1 per million births. And then the only effective therapy is liver transplantation. So because of this, we decided to treat this patient with gene therapy. This patient can survive with a, let's say, temporary treatment of phototherapy. So they have to stay there for 12, 14 hours a day just to lower bilirubin levels to life-compatible condition. So the first thing we did was just to create our animal model by introducing the mutation in the gene. And you can see here that the animals, the mutant animals, do not move as normal ones. They are jaundiced because you know that accumulation of bilirubin results in jaundice. So a yellow-orange discoloration of the skin and the white of the ears, of the eyes. And you can see here the mutant animals that reproduce most the major characteristic of the disease. So we started treating these animals with gene therapy, and we were able to save these animals that normally without treatment they die, and the treatment lasted for a long time. And since after that result, we entered in a bigger project that was related just to the treatment of patients. And so we use our construct containing our UGT cDNA under the liver-specific promoter. We improved our vector to try to reduce the dose and the risk. This worked very well. So we went into the patient. And the patient, what we can see here, I think that this is one of the best results I can show you, is that patients treated with this virus, so they reduced the amount of bilirubin that was present in blood, And the best example is the picture in which you can see the patient before and after the injection of our virus. And you can think this, if you think that 50 years ago, I mentioned that paper of Friedman and Roblin, in which treatment of modifying the function in a live organism was a kind of science fiction at that moment, and now we reach this point. So we are able now to modify the function of a gene. We are able to re-administer the missing function to cure a disease. And there are different examples, not only the one that I am showing you and the one of hemophilia that I have shown before, but there are other examples showing the potentialities of gene therapy today. But this is not all, because now we have other possibilities, and there is a kind of revolution today that is called the genome editing revolution. And this was the result of the studies done by Emmanuel Charpentier and Jennifer Doudnan, who were awarded the Nobel Prize a couple of years ago. But not only them, but also other researchers that I mentioned here, as Francisco Mojica, Feng Zhang, Dana Carroll, and George Church, who worked very, very hard to study this CRISPR-Cas9 system that was originally it is actually a kind of immune system of bacteria to prevent infection of particular viruses. And this was modified and transformed in a revolution tool that is now being applied not only in the laboratory, but also in the therapeutic side. So this has generated really a revolution in science today. So the idea is that with this enzyme, we can generate very specific double-strand breaks anywhere in the genome. And after this break, we can specifically modify any gene in the genome using this technology. So this is really a fantastic tool to do gene therapy. Before the existence of engineered nucleases, the possibility to modify specific sites of the genome exists, but it was really at an efficiency that was very far away from therapeutic purposes. And with the presence of this CRISPR-Cas9 platform and the other platform, we are now talking about the possibility, the therapeutic possibility to modify genes in the genome at our will. So this is what we are doing in the lab today. So we are modifying the gene just to introduce our therapeutic cDNA in hepatocytes. I'm not going to mention the details, but we are able to modify about, as you can see here, between 15 and 20% of the hepatocytes, and even higher in the lab now today. So this is something that is really very, very efficient. So we can exactly modify 20% of the hepatocytes in a very specific gene location to make this modify the hepatocytes to produce the therapeutic protein in which we are interested in. And when we apply this to our model system of Krigler-Najjar, obviously we were able, this was very, very efficient, and we were able to cure the animals. That untreated animals died very fast, while the treated animals survived with bilirubin levels that were really similar, exactly similar to the wild type animals. So this was really a fantastic result that we obtained using the system. But this is not the only thing we can do, because I will show you now in the next slide. So a very important application that is not from our lab, but I think that really it merits being told, because this is sickle cell anemia. The distribution in the world, you can see here the countries in which sickle cell anemia is more present. And the idea is that also for this could be used also, the same strategy has been used for thalassemia, beta thalassemia, in which we already know that there is a change in the formation of the tetramer hemoglobin molecule between fetal and adult. So the fetal contains the gamma chain that decreases production after birth because of the presence of this BCL11 factor that blocks the production of this fetal hemoglobin. And then hemoglobin beta is being produced. But in hemophilia, we have a mutation, in thalassemia, and in sickle cell disease, we have a mutation in the beta chain. So patients having this are sick. And what it was done, it was just using the CRISPR-Cas9 platform, we could direct this enzyme to destroy the enhancer sequence in which this factor BCL11 was bound. And after that, what happened is after treating the patient, you can see here in blue the production, the production of gamma globin, and these patients were treated. And importantly, you can see here, these are the bleeding, the transfusion events that before the treatment, you have very frequent transfusion events, and after the treatment, you have none, both from patient 1 and patient 2. One was for sickle cell disease, and the other was for beta thalassemia. In both cases, it was very, very efficient. So this is my last slide, so I would like just to conclude. So what I have mentioned is, let's say, a very happy end story in which we started from science fiction and we ended up in translation of this technology to the clinic. And this is what we have to do, as it was mentioned also in the previous presentation. So try to develop technology, and this technology has to be transferred to the public, to the population. So these treatments are really very promising therapies for genetic diseases, but not only, because I didn't mention, but these type of approaches with CRISPR-Cas9 are also being used for the treatment of cancer. And the treatment of gene therapy are very effective in adult patients, and there are different gene editing platforms that have been developed, and results obtained so far really are very surprising. And I am sure that in the next year, we're going to see many, many new applications of gene therapy in many different diseases. So with that, I would like just to finish. So to thank the people who participated, collaborators, and founded agencies. So I thank a lot for listening the talk and for the invitation again. So I am open for questions.
Muchas gracias por la excelente presentación, Dr. Andrés.
Okay, thanks for.
Hello, sir. I am Nalaka, one of the PhD students working with Professor Anil in the Maastricht University. So when I go through your website, I just saw that your group is working on AAV viruses on spinal muscular atrophy gene therapy.
No, not really. It's just I mentioned that, but because I think that these are, let's say, very important therapeutics that have been developed and mean very important to be mentioned, but we are not working in SMA.
Okay.
But in any case, if you want to ask me about SMA, I will be happy to try to answer your question. Don't worry about that.
Yes, sir, because like currently we are having a SMA population of 66 people we have, and we could identify the genetic mutations in only 22 patients. So 44 patients were negative for the major mutations. So there might be that what we think that there might be some point mutations or some other modifiers that will mimic the clinical outcomes of SMA, and we are looking forward to sequence their gene and identify these novel mutations. Sometimes those might be specific to our region.
Yeah, this could be possible. I think that your approach is correct. Just to try to sequence the complete gene, not only the main mutations. So Yeah, and then the idea will try to see if there is some therapy available. One of the problems that I didn't mention with this therapy so far is the cost of the treatment. That really is very, very high. And in most of the cases, if there is no, let's say, the health system of each country that is covering the cost, or some health insurance that is covering the cost, it is very difficult for the normal people to reach this therapy alone. But we also hope that over time, these kind of therapies will become more accessible to the public. In most of the cases, they are produced in a very, very low amounts, and the cost of research and the cost of production of the virus is really very, very high. And sooner or later, I think that this has to change.
Thank you, sir. Thank you.
We have time for one more question. Okay, in the absence of any questions, we will thank you, Dr. Andrés, for your excellent presentation. The molecular medicine is forefront of disease curing treatment strategies. Thank you very much.
Thank you very much for your invitation. I'm happy to if you have any other question, I just send me an email, and I will be very happy to answer it. Bye.
Thank you, Professor. So we want to the speaker. So with great pleasure, I like to introduce our third speaker of the session, Professor Lyudmila Turyanska, who has joined online. Professor Lyudmila is from the Center of Additive Manufacturing at the Faculty of Engineering, University of Nottingham. The research focuses on the development of novel low-dimensional materials and hybrid low-dimensional composites for applications ranging from optoelectronic devices to other healthcare technologies. She's involved in integration of novel functional materials with additive manufacturing technologies for device applications. So, ladies and gentlemen, please welcome Professor Lyudmila.
Hello. Sorry, my name is Oleg Makarovsky, and I'm here on behalf of my colleague Lyudmila Turyanska. She sends sincere apologies. Unfortunately, due to some emergencies this morning, she cannot be with us today. She sent the video of her talk, which I think will be played now in front of you, and I'm here to answer your questions after the video.
Yeah, please go ahead.
Good afternoon, everybody, and thank you very much for giving me this opportunity to present our work to you. It is my great pleasure to be able to be here, albeit online, and tell you a little bit about our work on additive manufacturing with functional materials. So first of all, I would like to introduce our center to you. So we are based in the Center for Additive Manufacturing at the University of Nottingham. The center was established in 1992, so it's about 30 years old, and we began our research into additive manufacturing only in 2000. So more recently, we started looking beyond that structural additive manufacturing, more into functional additive manufacturing. And there are lots and lots of opportunities that can be opened up with that direction, out of which the devices and healthcare applications are those we're carefully looking at. So as a center, we are a group of about 10 academic members of staff and another about 100 research staff and PhD students, all of whom are dedicated to research into additive manufacturing. So we are working across the disciplines, and we have members of team who are physicists, engineers, pharmacists, chemists, biologists, and it is ever growing and ever expanding research. So we have a number of different activities, all of which are supported by our Engineering and Physics Sciences Research Council. So we're looking at formulating materials for the future of additive manufacturing through our program grants, through platform grants, but also we are actively involved in training new generation of researchers, new generation of PhDs. More recently, we have also developed a new and dedicated master's program. So that's a 12-month program where we have focused research-based modules developing knowledge of additive manufacturing, developing the knowledge of requirements, limitations, applications, etc. So if any of you are interested in any of those opportunities, do feel free to get in touch with me. But generally, we are very well equipped with a number of different additive manufacturing facilities. That is from powder bed fusion to VAT polymerization, going all the way from that bulk printing, bulk deposition to nanoscale deposition. We are supporting the development of those facilities with the companies, but we are also developing our design and computational methods to predict the properties of the structures that are being produced. So in our computational methods, we're particularly interested in looking at, for example, at metal jetting. You might all be aware that additive manufacturing is heavily used in aerospace industries, in automobile industries, and there the weight limitations or the weight requirements are very, very strong. So we have a very strong team looking at developing modeling of the lattices and topology optimization, looking to reduce the weight of those printed particle parts and yet to retain their mechanical properties or even improve those properties. In terms of materials that we are developing and we are working with, we go really across the range of materials available, from polymeric materials to magnetic materials, metallic materials, but also we are looking at that frontiers research where we develop biomaterials, where we develop low-dimensional materials for additive manufacturing of devices, which I will tell you a little bit more about. Really, the devices is quite a broad definition, and devices are used everywhere, and that is from 3D electronics, for example, wearable electronics that is becoming very, very popular, all the way into the electronics that are used and developed on curved substrates such as satellites, but also looking at that healthcare end where we are integrating function, for example, tactile function or sensing into prosthetics, so to be useful for healthcare applications. Our recent development, again, highlighted opportunities of additive manufacturing in pills printing, so in printing of pharmaceutical agents. But in this work, I will tell you a little bit more about our work on low-dimensional materials, and hopefully that will be of interest to you. So you might know that the definition of low-dimensional materials is quite broad, and indeed we're just looking at the materials where in one or more direction the motion of the electron is confined. That gives us the tunability of their properties in terms of their optical properties, in terms of their electrical, magnetic properties, etc. So one of the more interesting materials that attracted a lot of attention recently are perovskite nanocrystals. Some of you might have heard about extensive research into using perovskite nanocrystals into photovoltaics. So for energy harvesting with the aim to improve the performance of solar cells. There is still a number of challenges that we are facing with these materials, and one of them is their stability. So as researchers, as material researchers, were looking to improve their stabilities. And one of the directions that we have explored is looking at all inorganic nanomaterials. We all know that inorganic crystals are more stable than those that contain organic component. So we are working with the nanocrystals that are based on a metal. In this case, it is lead, cesium, but we're adding halide as a non-metal component. And by tuning the halide composition, so that's iodine, bromine, and chlorine, we can change their properties. So the way we address their stability is by improving the surface of those nanocrystals. So once the crystal is growing, we know that the surface to volume ratio is largest for the smaller nanoparticles. So the properties of the surfaces are really detrimental to the overall performance of these nanocrystals, of these nanomaterials. And we are looking at improving that surface. So we can achieve with these nanomaterials the tunability of their properties all the way across the electromagnetic spectrum from blue into red, as you can see on this color gamut. One of the important as well aspects of these nanocrystals is the ability to produce the materials that have very high quantum yields, reaching close to 100%, so close to unity. And that is very desirable for a number of different applications in optoelectronics. So one of the approaches that we have explored aiming to improve the stability of the nanocrystals is the choice of so-called capping molecules, choice of molecules we're using to passivate the defects on the surfaces. And by using those materials that have functional groups that allow stronger binding, we can passivate the surface defects. By using some of the materials that prevent the accessibility of moisture to the surfaces, we can also improve the stability of these materials. And in this work, we have demonstrated that the very stable nanocrystals can be produced with the emission from green to red by changing their composition. So you can see that these are iodine-based, have red emission, and bromine-based have blue emission. More recently, we looked at integrating chlorine as well, and we produced some blue-emitting nanocrystals. So the quantum yield of these nanocrystals was over 90%, which is absolutely brilliant achievement, and we had them stable for up to half a year after synthesis, which is as well quite a significant achievement. But colloidal stability is important, but more important is the stability of these nanocrystals in devices. So we have explored the stability of light-emitting diodes based on these nanocrystals. In this particular example, in this particular work, we have used halogen-based capping molecules. So that halogen that is present in the capping molecule can integrate into the crystal lattice, making the devices very, very stable. And we have indeed achieved very good performance of those red emitting LED devices that were much improved by integration of those halogen-based ligands. So really our chemistry-based approach to improve the surfaces does work and improve the stability, not only the shelf life of the nanocrystals, but also the stability of the LED devices. So in this case, we have produced device using solution-based processing for all the layers. And we still have that organic component as a capping molecule in these nanocrystals. So more recently, we have looked for opportunities to produce all inorganic, but highly stable and highly efficient nanocrystals. And in this so-called core shell approach, we have achieved a core, a fully inorganic core that epitaxially capped with another layer, and by changing those layers and by carefully controlling their composition, we can avoid the lattice strains and we can passivate the defects of that core. And it is that core material that gives us the optical performance. You might know that this is the approach that was used quite a lot in semiconducting quantum dots and that have enabled their use in optoelectronics, in television, for example. So we have adopted that sort of direction and produced for the first time this core shell nanocrystal, perovskite nanocrystals with very, very high quantum yields and with very stable performance. In this case, you can see the LED operating in red wavelengths of electromagnetic spectrum. Again, we have significantly improved long-term stability in these nanocrystals, but also the device performance. So color tuning is a very, very big challenge, and you might be aware that the detection and emission in the wide spectrum is still quite challenging because all our white LEDs still have a mixture of different colors and some preference for a particular color range of electromagnetic spectrum. So one of the approaches that can be enabled with perovskite nanocrystals is to achieve more precise color mixing. So what we have done in this case is we've used our stable nanocrystals. We have integrated them with the LED that was produced using conventional processing. So we just took a conventional gallium nitride diode, those blue LEDs that we are also familiar with, and we have surface decorated them with our nanocrystals with emission in green and red. And by mixing the ratio of those materials, we have produced the white LED photograph of which you can see here in the middle of the picture, right? Then in this work, we have looked at stable performance and a tuning of the color with the mixing of those components. So lots and lots of flexibility with these materials in this sort of application, but looking also at not only large LEDs, but also micro LEDs, which are in demand for electric circuits and for more advanced devices. So the photoresponsivity, so that's the light emission, but light detection gives us also quite a lot of interesting challenges, which we were able to address with these nanocrystals. And in this particular case, what we have demonstrated is that our stable nanocrystals can be used as a photosensitive layer to decorate graphene devices. In these device geometries, we are using graphene as a conduction channel. We are putting our optically active nanocrystals on top of these devices, and we can achieve the photoresponsivity up to 10 to the 7 amps per watt, which is one of the record high responsivities reported for this type of devices. We have very reproducible on-off times, and we are looking at improving this performance. So more in controlling the time of this response is controlling the wavelength range of this responsivity. As you can imagine, having the nanocrystals optical properties tunable by their composition, as I have showed to you on previous slides, gives us opportunities to have a detection tunable by the composition, and we have indeed demonstrated the sensitivity, photosensitivity of this type of detectors across the visible range from blue detection to red. Now the sensitivity and the opportunities for these devices is really fascinating, as you have seen, and we are looking to harvest them using additive manufacturing technologies to have the devices that can be easily produced with large numbers as well. And in this space, we have developed quite a lot of additive manufacturing, or we adopted quite a lot of additive manufacturing technologies for these materials. we have formulated these materials for the deposition using these technologies, and we are looking in terms of applications, well, across the range from radio frequency sensing to photon detection to displays. So one of the exciting opportunities with this, again, with integrating these materials with additive manufacturing, is the possibility to produce flexible materials. We are looking at devices that operate on flexible substrates, as you can see on this image on Kapton, on fabrics, which we have demonstrated as well, and we can print them with the scales looking all the way across the ranges from micron sized to centimeter size, as you can see here in this image. So in this particular work, we have demonstrated that a number of different nanomaterials, from graphene to hexagonal boron nitrides, these are the group of two-dimensional materials, to Silva nanoparticles that are zero-dimensional materials, can be integrated within one device, and we have managed to produce this fully 3D printed graphene transistor with the HBM gate, top gate, for the device performance, and Silva nanoparticle produced electrodes. The performance of this device was quite good, and we have done some extensive studies to understand how these materials perform in these heterostructures. So as an example here, you can see that the device performance can be interrogated to quite an extensive detail with time of flight mass spectroscopy used to map in a 3D to map the composition of these devices. So you can see well-defined top layer, so that's the hexagonal boron nitride. We can see well-defined silver printed. We can see some intermixing between the silver and the graphene in these devices, but we can now control that intermixing to our benefit to have better performance in those devices. So understanding how these printed materials can be integrated with other materials, as I have shown to you in LEDs, we have also looked at using printed graphene as a contact material for exfoliated 2D materials such as indium selenide, for example. And in this image, you can see the device. So we are again going across the manufacturing technologies from exfoliation and stamping to additive manufacturing. And we have produced photon sensors with a stable and very sensitive performance across the wavelength range of absorption of these indium selenide flakes. And those of you working with the two-dimensional materials will know that producing ohmic contacts to the exfoliated materials is quite a large challenge. So using this additive manufacturing of graphene, we have addressed that challenge, and we have demonstrated a very good performance in those devices. So now, thinking about what else can be done, you can imagine that lots and lots of opportunities for integration of other materials, such as LEDs and photon sensors, which we have managed to fully inkjet print, fully 3D print using perovskites integrated with these technologies. But we are also looking at that multiple functionalities, because we have now developed so many materials. We can look at one fully printed device where we have temperature sensing, pressure sensing, humidity sensing, as you have illustrated here on the right-hand side with graphene sensors, but we have now integrated also optical sensing and chemical sensing into those devices. So understanding the charge transport through those networks presents quite interesting fundamental tasks, which we are working with our physics colleagues to develop quantum mechanical models that explain those devices. So if you're interested in any of that, you can read our papers, or please feel free to send me an email, And I will be very happy to talk to you about that. Right. And then a few more devices that we are looking at are those antenna, as I said, or inductors. And you can see a couple of those designs that we have managed to reproduce, both with the planar printing, with heterostructures, but also with more 3D style designs using metal nanoparticles as shown here at the bottom right corner. So there is a lot of different opportunities for us to go through, and we expand our work more to look at such structures as metamaterials, to look at multi-material printing at a nanoscale, as illustrated with this work with the surface decorated gold particles. So here we have produced structures with a submicron resolution, and we have managed to integrate very selectively metal nanoparticles into those structures. So I'm running a little bit out of time, and really a lot I wanted to tell you in this overview talk. So I hope that I managed to excite you, to present to you the opportunities that can be unleashed, that can be opened by integration of functional materials with additive manufacturing technologies. So the work that I have presented is really the work of a team where we have good researchers working on different aspects of this integration. And I would be very happy to answer any questions you might have. So do feel free to get in touch with me by email if you're not able to ask the questions in this talk. And with that, I want to thank you very much for the attention, and I will be happy to answer any of your questions. Thank you.
Yeah, thank you very much. Yeah, so thank you very much. We are moving on to the next presentation. Thank you very much.
Thank you.
Yeah.
It's a great pleasure for me to introduce the fourth speaker, Senior Professor Sagarika Ekanaik. Professor Ekanaik is chair at the Department of Biochemistry, Faculty of Medicine, University of Srijajanapura. She obtained a PhD in Food Chemistry and Applied Nutrition from Lund University, Sweden in 2005, contributed to the food-based dietary guideline development for Sri Lanka, very important task in 2012, and been in the expert panel updating the FBDD in 2020. She has authored more than 75 publication peer-reviewed journals, produced over 20 research communications, and contributed to the book chapters and textbooks. Out of many esteemed posts held throughout her career, President Institute of Chemistry, Ceylon, 2021, President Section A, SLAS, 2012, Chairperson, General Research Committee of the SLAS 2017 are the highlights. Senior Professor Sagarika Ekanaik is a recipient of over 40 awards, honors for research, including prestigious Dr. C.L.D. Silva Gold Medal in 2017, Professor M.U.S. Sultan Baba Award for Research in Advanced Chemistry in 2008. And there are several presidential awards for research excellence in 1999, 2003, 2006, 2007, 2009, 11, and 14. And a lot of merit awards for scientific publication. And Vice Chancellor's Award in 2016, and many fellowships from the international and local agencies. So we have experts in the field for your attention. Let's welcome Senior Professor Sagarika.
Thank you. Thank you, Professor Nimal, for that very long introduction. I hope I'll be able to do justice for that introduction. The plenary chairs, academic members who are present here, the coordinators and conference organizers, good afternoon to and all the presenters and people who are joining online. Good afternoon to all of you. I would like to take this opportunity at the beginning to thank the co-chairs of this session for the invitation given to deliver this plenary lecture. So my story is not going to be as fascinating as Professor Nalin's, because I'm going to touch something which is very close to your hearts, food. So it's actually a very small part of the work that we have done, but I thought it is good to elaborate so that you all can take home some messages with you, as the topic I'm going to talk about is very much closer to your hearts. And I'm sure you will have people who can be helped with what I'm going to say. Right. So thank you again for the opportunity. And it's nice to be back at KDU, because I was here at the medical faculty lecturing the early cadets, which was very enjoyable. But as Professor Nalin said, the discipline, I had a problem because they were falling asleep during the lectures most of the time because they were tired. So we had to actually ask the hierarchy to stop working these children so much early in the morning, because they were so tired when they came for the lectures. But they were a well-disciplined lot, I have to say. Right. Okay, to move on with my lecture, I hope I'm not taking much more time. So my topic today is Sri Lankan traditional rice and foundation for a healthy diet. So my pictures are not coming, I don't know why. So I hope that all of you will be able to take home some good messages after this presentation. Right, so the overview of my presentation is basically about rice consumption patterns in Sri Lanka and the processing of rice. Also a little bit about the reasons for resurgence of Sri Lankan traditional rice cultivation and consumption, and a little bit of a comparative analysis of the different processing methods of traditional rice varieties and how these processing will have affected the nutrient content and the glycemic response of traditional rice varieties. So moving on. Yeah, my pictures are not coming. I had some maps here. I don't know why. Right. As all of you know, rice is the staple food of more than half of the world population, especially in Asia and also in some parts of Africa and also some countries in South America. Also for us, rice is the most staple food, and also the food that we consume, majority of consume more than twice a day. And look, if you look at the per capita consumption of rice and the rice-based products, it's about 114 kilograms per person per year. So that's a lot of rice that we consume. So these rice, the rice we consume gives us the major portion of our energy, about 40 to 70%, and also, very importantly, 1/3 of the protein requirement of a normal Sri Lankan average person. In addition to that, rice also provides us with a lot of nutrients, minerals, vitamins, and also rice contains phytonutrients which are bioactive, as I'm sure some of you here have already done research on and maybe you are aware. But we have to remember that whatever the rice we consume, the nutrients in rice will depend on the processing the rice is being subjected to. So what are these processing techniques? I'm sorry you all can't see any of my pictures. I don't know the reason, but I'm in trouble if I can't show them my pictures because I have a lot of graphs. Right. Until you fix it, I will try to continue. So rice is actually available as raw rice, as I'm sure all of you all know, which we know as kakulu rice. That is just the D hull. You just remove the husk from the paddy. Then also we have polished rice. Also, we have raw polished rice, as you all know, kakulu polished. So that is outer covering of the rice is removed. And then also, but remember when you remove the outer covering of the rice, you are getting rid of most of the nutrients in rice. Thank you. Right. So all constituents in rice except carbohydrates will be removed when you go for extensive polishing of rice. Also, rice is available as parboiled rice. I'm sure all of you all have heard about Tambapuha rice. So this parboiled rice due to the parboiling process, retains most of the nutrients in the rice grain. But as we all know, parboiled rice is not very popular among the younger generation. They don't like to eat parboiled rice. But remember, this rice retains most of the nutrients. So going back in history a little bit, in 1960s, Green Revolution with the expulsion of population led to introduction of many improved varieties, which we are consuming now. And this actually, they were high yielding. So because of that, they became very popular to feed a lot of people. Then this actually led to a decline in the Sri Lankan traditional rice varieties, I think over 2,000 varieties, which we have been consuming for over thousands of years. So also, as you can see from this graph, in 2060, after the introduction of these newly improved varieties, with the development of technology, as mentioned by Professor Nalin in his lecture, more technologies were developed and rice were more refined. When the rice came into the market, they were more refined. So we became used to eating more refined rice rather than parboiled rice or the whole grain rice, as we call raw rice, just behulled rice. So as you can see, but from 2016, there has been a decline in this refined rice consumption in our country. So as you can see from this slide, coming back to the future, the high prevalence of non-communicable diseases, which are nutrition related, are partly attributed to this consumption of highly refined rice. In addition to that, you all know we have become more sedentary in our lifestyles, and also we are not taking enough adequate vegetables and fruits in our diet. So all these have contributed with consumption of increased refined rice to this high prevalence of non-communicable diseases in our country. So this is why the traditional rice varieties have made a comeback, and as you all know, according to indigenous medical practices and also Ayurveda, these traditional rice varieties are supposed to have good nutrients as well as they have medicinal properties. So in Ayurveda and indigenous medicine systems, these traditional rice varieties are being used in a variety of ways as porridge, as extracts with herbal extracts, etc., as remedy for a variety of ailments, including malnutrition. So in addition to that, I think more importantly and related to your theme of the conference, this traditional rice farming is said to be environment friendly and also economically viable and sustainable, because these traditional rice varieties can withstand all the different biotic stressors, pests and diseases. In addition to that, traditional rice varieties are also grown under normal environmental conditions. So we don't have to use a lot of fertilizer, etc. in growing traditional farming. So it is sustainable. So the comeback of traditional rice varieties, consumption and also increased cultivation after about 2016 is mainly due to their beneficial effects. And you can see these traditional rice varieties now being available in markets, and there are many outlets selling these traditional rice varieties, especially in the urban areas. So the interest is again due to this health and nutritional benefits of traditional rice varieties, especially in relation to controlling the blood glucose levels and also reducing the non-communicable disease incidence. Right. So moving on to traditional rice varieties, nutrients. Much work has been done by many researchers in Sri Lanka. We have to give credit to them. They have worked on traditional rice varieties, numerous varieties, and have put out data on the proximate composition, the nutrient composition, and also bioactive compounds of traditional rice varieties. And some of them are rich in protein, more than wheat, and some of them are very rich sources of essential minerals like zinc and iron. So credit goes to the researchers who started all this research. However, all these data are basically related to only one type of rice, that is raw whole grain without polishing or parboiling. And also most of these data are on uncooked rice. But as all of you know, we usually eat rice after cooking. So we need to know what are the effects on these nutrients and also the parameters related to reducing the non-communicable diseases after cooking. So that is what we actually did. So that is what I'm going to explain to you, a small part of the work that we have done. So I hope you all will be taking home some messages after this lecture. So it was important, since we noticed this fact, this gap in the research, it was important for us to scientifically validate why the popularity of Sri Lankan traditional rice in the current context of high prevalence of non-communicable diseases, and also the different processing techniques which will help us to identify the processing that would be most suitable in for people who have these non-communicable diseases, or even for normal people, because we develop into a non-communicable patient if our dietary patterns are not good enough. So there was comparative analysis. We did a comparative analysis of using 6 different types of traditional rice varieties. They were differently processed, like raw rice without polishing. Then we had another fraction, a raw polished rice fraction, and another fraction, a parboiled fraction. And we cooked all of these differently processed 6 different rice varieties. We cooked, and then we studied their nutrients and glycemic response. So as you all can see from this graph, this is why I said if my pictures are not there, then it's not going to be of any use. So this graph shows you the 6 different rice varieties, the 3 different processing methods, and the digestible carbohydrate content in freshly cooked rice. That is the rice that you eat in your plate, not rice flour, or not uncooked rice, but this is cooked rice in a plate. So as you all can see from the ash bars, the parboiled rice contain the least amount of digestible carbohydrate, whereas raw unpolished rice and polished rice contain higher amount of digestible carbohydrate. So we were wondering why. Then what we looked at was the moisture content after cooking. So when we looked at the moisture content of cooked rice, parboiled rice had more than 75% of moisture in the cooked rice. So compared to raw polished or raw unpolished rice of same varieties. So this actually showed us that when you are eating parboiled rice, the carbohydrate load that you are taking in is much less compared to raw polished or raw unpolished rice. So this is one reason why parboiled rice is good for people who want to reduce their energy intake, also to reduce their carbohydrate intake. So the protein, when we looked at the protein contents of these 6 different varieties, differently processed, the protein content in all varieties, all differently processed methods was not very high. It was 4%, less than 4%. But as you can see, due to the high moisture, the parboiled rice content less protein content compared to other 2 processed methods. But then again, note that it is just a small difference, because unlike carbohydrate, the protein content in all these varieties was less. Therefore, this difference is not felt very much when you take the protein content. The fat content, when we looked at the fat content, fat in all rice was low. As you all know, rice is usually a low fat food. So when we looked at the fat content of cooked rice portion that you consume, whole unpolished rice had highest amount of fat among all varieties. Then parboiled rice had a same amount of raw polished rice, despite parboiled rice having high amount of moisture. So if you look at it in a comparative way, parboiled rice would have more dietary fat than raw polished rice. Then we looked at dietary fiber. So because it is important in the context of NCDs. So when we looked at dietary fiber in a cooked rice portion, raw unpolished rice had the highest amount of dietary fiber, followed by parboiled rice and raw polished rice. So you know, when we polish, we remove the outer covering, which has the dietary fiber. So we get rid of the dietary fiber. So raw polished rice contains the least amount of dietary fiber. However, when we compared cooked and uncooked rice, cooking increased the dietary fiber content. So I think this has not been reported earlier. So this is something we observed. Dietary fiber increased in all different rice varieties, irrespective of processing. Right, I am showing you the graph below. This is on rice flour actually, just to illustrate the difference clearly, because then you all can see the difference. So this is the uncooked and cooked 6 different rice varieties subjected to different processing methods. So as you can see, uncooked rice, all varieties contained less dietary fiber, but cooking contributed to an increase in the dietary fiber content, with the highest increase in parboiled rice and unpolished raw whole rice. The same was observed after cooking. Then I think you all have heard about resistant starch. Resistant starch is undigestible starch that is produced when rice is processed. So unpolished rice, raw unpolished rice, and parboiled rice contains similar amount of resistant starch, with raw polished rice containing low amount of resistant starch. So cooking, once again, as you all can see in the graph, increased the resistant starch content in all different varieties, with parboiled rice showing the highest increase. So why we studied resistant starch? Because it is evidenced that foods containing high resistant starch is good for controlling the glycemic response. So when we looked at the glycemic responses and the glycemic indices of these varieties, as you know, carbohydrate is basically related to giving rise to a glycemic response. All of you are aware of that. So if you control your carbohydrate intake, the quality and the quantity both, then you can control your energy and the glucose intake, and thereby control the development of these nutrition-related non-communicable diseases. So basically, glycemic index is a quality index of carbohydrates, because glycemic index will tell you whether the carbohydrate is digested quickly and absorbed quickly and gives rise to a higher blood response, or whether they are digested slowly, absorbed slowly, and gives rise to a lower glycemic response. So it's a quality parameter. So when we determine glycemic index, what we do is we give a portion of rice containing the same amount of carbohydrate. So the amount of carbohydrate in all food portions is the same. So whatever the difference that will come due to the highly digestibility or low digestibility of the carbohydrate. So foods are categorized based on their glycemic response as high, low, and medium. Then we have the glycemic load. Glycemic load is the glycemic carbohydrate content in the food portion that you eat. This is calculated using glycemic index. We calculate glycemic load because sometimes the portion that we give to eat to determine glycemic index is large. 50 grams carbohydrate portion in some foods is very, very large. So to apply the glycemic index concept to an edible portion, we use glycemic load. So dietitians can use the glycemic load value when you have to prescribe diets to your patients. So this again is categorized as high, low, and medium. So low GI foods are basically considered to be good for many ailments. As you can see, there are many, many factors that by consumption of low GI foods will contribute to. So in addition to that, many scientific organizations, dietetic associations, including Nutrition Division in Sri Lanka, also promote consumption of low glycemic index foods if you are suffering from non-communicable diseases or if you want to maintain good health. So this is what we did. We took these 6 rice varieties, differently processed rice varieties, and we determined the glycemic index because we thought that will validate the health benefits and the claims why traditional rice varieties are making a comeback and why people are consuming these varieties. So these are the portions that we gave to determine the glycemic index, and all these rice portions contained 50 grams of digestible carbohydrate. But you can see the volumes are different based on the carbohydrate. So these are the glycemic response curves after consumption of these varieties. So the green lines at the bottom in all 6 varieties actually. I did not have enough space, so I have put only 4 graphs here. So the green line at the very low curve is for parboiled rice. Professor Charitha, can I have 2 minutes? Right. So then the orange line is actually the raw polished rice glycemic response. The blue line on top is the raw polished rice response. So you can see parboiled rice gives a lower response and polished rice gives a higher response, and unpolished whole grain rice gives a medium type of a response. So using these values, we calculated the response was same for the other 2 varieties also. Red line is actually glucose. So when we calculated the glycemic indices of these varieties, I will first take the green ones. Parboiled rice elicited low glycemic response. Then raw polished varieties, that is the ones that we like to eat, they gave a higher glycemic response. Comparatively, raw unpolished rice, the whole grain rice, elicited medium glycemic index values. So glycemic load values given in the second line are for 50 grams of digestible carbohydrate. But then the people found that the portion we gave is too much to consume. So when we back calculate the amount of glycemic carbohydrate in an edible portion, parboiled rice elicited a lower glycemic load compared to other 2 differently processed varieties. This is for the red varieties we studied. The same observation was made for these other high glycemic index, low glycemic index, and low glycemic load of parboiled rice. The same was observed for the white varieties, the 2 different white traditional rice varieties. So just to illustrate, these are the portions we gave to determine the glycemic index. Each of these portions contained 50 grams of digestible carbohydrate. So as you can see, parboiled rice, the portion was really huge. People could not consume this portion because parboiled rice contained high amount of moisture after cooking. So this is why the glycemic load becomes lower, because the portion size is large when you take cooked parboiled rice. So this graph actually tells you the portions that we gave to determine the glycemic index. They are huge, as you can see here. For the 3 differently processed varieties, parboiled rice had the highest volume of rice, which contained 50 grams of digestible carbohydrate. So when we bring it back to an edible portion, the glycemic load became lower. So if you take parboiled rice, glycemic index is less due to high resistant starch and low high dietary fiber, and also they contain high amylose, and glycemic load becomes less because they absorb a lot of water after cooking. Therefore glycemic load becomes less and less than raw and raw unpolished rice. So coming back to give a summary, parboiled rice to a great extent, and also raw unpolished traditional Sri Lankan rice varieties to a lesser extent, irrespective of variety, are nutritionally and health-wise more superior compared to raw polished rice varieties, and also they are much more suitable as a foundation for the rice meals. So these varieties, basically the parboiled and the raw unpolished varieties, can be recommended for people who want to control their energy intake and also the glycemic load. In other terms, people who are suffering from non-communicable diseases. So my final slide, in fighting high prevalence of non-communicable diseases and also in improving the health in general, the health of people, and contributing to sustainability, Sri Lankan traditional rice, especially the parboiled and raw whole grains, could play a significant role. So I have to thank my MPhil student, Ms. T.A.P. Yuthennakod. What I presented today is part of her work, actually. She's currently pursuing her PhD in UK. And also we have been, unlike the development of gadgets, we have been able to contribute to the development of Sri Lankan policy at policy level with this research to develop food-based dietary guidelines of Sri Lanka. So I think that is a small step that we academic have been able to do by contributing to a policy level. So I'll take leave of you with this saying, which was said by Hippocrates in 400 BC, which I think is applicable to today also. Thank you very much for your attention.
Thank you very much, Professor Sagarika Ekanayaka, for very important presentation. So what is the take-home message? Any young lady could answer? So you are going to be, no, you, only the male also has to cook, but you also are contributing in larger, in the kitchen. So the parboiled matter, we can interpret as nadu.
Nadu, yes.
So that is how
Even
people know
improved
nadu.
by
So
varieties.
people are away from nadu, they want to get samba or other sambas for the high level of influence in Sri Lanka. So go for those things, no? Kiri, samba and all that. But nadu is the best. Right? Because now in the Sri Lanka, the diabetic population is increasing by the day, especially the young children, the type 1 diabetes. From the Lady Ridgeway data, it was found that there's a large number of type 1 diabetes are increasing. IBDM are increasing every year. A lot of children are coming to the pediatric endocrinology clinic in the LRS. So because mothers are feeding them with rice, all this fried rice and all, they're fond of it. Other additives with polished rice. For your young ladies, you can bring up your children with nard. So that is the advice. Any more questions? Any questions from the audience? So I have asked one question already.
Madam, thank you very much for that. Thank you very much for that actually eye-opening presentation. Just a request and a small question. Request is, as our co-chair, Dr. Punyasiri said, Professor Punyasiri said, why don't you, publish this data in a late to our normal people? Now you have presented here, and I'm not sure whether you have already published it, because the problem, yeah, the problem is, madam, it's not coming for our society generally. Maybe published among our scientific society, but not for the general public. So I request you, I'm sure now you are very much familiar to do those things. I know that because you are a very good teacher. As today also we all understand what you said. So you can always do it. So just request you to publish this in the late term and to make eye open for general public. And my question is, madam, when you preparing rice in different ways, now you said that you have checked in different ways. I mean, some are using, now in single terms, we get thoroughly pay. Some are rice cooker. Are there any difference when you cook in those different places?
Yes. We did a study. We actually did a study using basmati varieties, if I remember correctly, some time back. We used the rice cooker and the microwave. So microwave cooking, I think there was a slight decline. But when we talk about GI, the slight declines are not so very effective, because we take the average of 10 people usually. So the standard deviation is there. So these small deviations falls within the standard deviation. You don't get a big difference. So I don't think if you cook rice too much, like berry rice, if you like to eat, then that is not very good, because then the starch becomes gelatinized and it is easily available for digestion. Then of course that can make a difference, because if you take kiribat, we have done some work with kiribat. Kiribat had very high GI. That is because you cook it, and you make it into a mulch. So then the starch becomes very much available, and then you can, it can give rise to a high glycemic response. I think with the parboiled rice, that does not happen. I don't know whether you all are familiar, but try to cook parboiled rice. It's very difficult to make it a berry rice. It never happens. That is because of the previous processing rice has undergone. So because of that, the parboiled rice, the starch becomes resistant starch, part of it. And also whatever the starch is available is a little bit, I think, not much available for digestion. It gets digested very slowly. That is why we see a lower
I have a questions from the madam a long time back. When I came as the thesis examiner for the job.
Yes, what I do is I try to give as much as possible. The thing is, any rice is not only traditional rice varieties, but any parboiled rice is good enough if you want to control the energy intake and the any parboiled. So thank you. I will try to do that.
Thank you very much, madam, for the excellent presentation. So we can conclude all the presentations. Yeah, over to Avan. That's one more presentation.
So, yeah. The last presentation, last speech for today's session is by Professor Eric Hoffman, who is joined online. Professor Eric Hoffman is the Associate Dean, Research and Development, School of Pharmacy and Pharmaceutical Sciences, Binghamton University, USA. Prior to joining Binghamton, Eric Hoffman was Assistant Professor of Neurology at Harvard Medical School and Children's Hospital, Boston, Professor of Molecular Genetics and Biochemistry at the University of Pittsburgh School of Medicine. And also he had been the Director of the Center for Genetic Medicine at the Children's National Medical Center and a Professor and the Chair of the Department of Integrative Systems Biology at George Washington University. Throughout his prestigious career, he has discovered dystrophin, the protein product of Dutch muscular dystrophy locus, earned 21 patents and published about 600 papers with about 58,000 citations. Professor Hoffman is a flagship personality in translational research in neuromuscular diseases. He has co-founded 3 companies that spun off from his efforts, namely Troins LLC, Revragen Biopharma, and AGADA Biopharma. So I think it's time to listen to Professor Hoffman's speech. Over to Professor Hoffman. Over to you.
Thank you very much. Can you hear me okay?
We can hear you well, Professor Hoffman. Please go ahead.
Thanks. I first like to thank my colleague and friend, Dr. Neil De Silva, and Vice Chancellor MP Peres for the kind invitation to speak to you today. I have a broad task here, so I'll try to move this along. I'll first speak to neuromuscular banking, which we can define as the long-term storage of biological materials from neuromuscular patients for research or molecular diagnostic purposes. And typically that's either DNA or blood, from blood, and that enables companion diagnostics for precision therapeutics, increasingly common in both cancer and rare disease, genetic disease. Gene discovery, genetic linkage studies, mutation studies by sequencing, and tumor sequencing. And then pathological tissues for messenger microRNA or protein studies. And that really helps get at molecular pathophysiology. So Duchenne muscular dystrophy as one example, the mutation initiates the process, but then the disease and the disability is due to downstream events from that primary defect. So using the tissues from the patients, you can answer questions as to what actually causes the symptoms of the disease, even though we know the initial root cause. So the primary defect you can get at from DNA, but downstream consequences from pathological tissues. So I thought I'd first give just one example from my laboratory where we used pathological tissues that we had banked from muscle biopsies from patients from a molecular diagnostic service. And I ran a molecular diagnostic lab for decades and had thousands of both DNA and muscle biopsies frozen at that I could use for this research. So from these DNA, you can identify new genes, mutations, new methods, and pathological tissues get at the pathophysiology. So the disease I'll talk about first is laminopathy. So these are disorders of the nuclear envelope. So the double lipid membrane that encapsulates the nucleus and all the DNA. And there's a series of proteins around the nuclear envelope. And one protein in particular, lamin AC, is a key structural component. But increasingly, diseases or disorders associated with single amino acid changes of this gene now have been shown to cause premature aging, peripheral nerve disease, muscular dystrophy, partial lipodystrophy with loss of fat deposits, and cardiac problems with cardiac myopathy and conduction defects. So the same gene, different amino acid changes cause all these different symptoms. So we wanted to ask this question: why do different single amino acid changes lead to such dramatically different symptoms in the same gene? So this is just a schematic of the nuclear envelope and the nuclear envelope around the nucleus and the double membrane bilayer with the proteins going through it. But the key thing I'll address is that mutations of lamin AC, I'll show, change how DNA is bound to the nuclear envelope. Because increasingly we recognize that attachment sites for DNA or chromatin at the nuclear envelope become inactive or heterochromatin, whereas regions that are not attached to the nuclear envelope go into the center of the nucleus, and those become more transcriptionally active, and that's called euchromatin. So our hypothesis was that lamin A is a problem with cell commitment during differentiation because of changes or abnormalities in how the DNA attaches to the nuclear envelope in specific cell types. So we thought that perhaps in muscle cell types, as a cell goes down the muscle lineage, then mutations that cause muscular dystrophy change how chromatin attaches or detaches from the nuclear envelope, whereas different mutations change how, say, a lipid cell goes down differentiation. So we engineered two different mutations. These were just 10 amino acids apart. Both of them were normally arginine and were changed to tryptophan in these patients with muscular dystrophy or lipodystrophy. And then what we did was engineer, and I'll describe just briefly the results from this publication where we defined this problem. So the goal is to see if keratinocytes and myoblasts are going down epigenomic pathways, if these mutations change these pathways. So how we did that is we took each of these genes and mutations and fused the protein to a bacterial protein called DAM, which causes methylation of adenines. So eukaryotes, humans, do not methylate adenines. We only methylate cytosines, and cytosines methylation is what changes euchromatin to heterochromatin. So by doing this, we could see where lamin was contacting DNA because its attached DAM methylase would be methylating adenines. So then by doing this in the whole genome in specific cell types like myoblasts or adipocytes, we could see how during development, lamin A was interacting with different regions of the genome through detection of these novel methylated adenines. So when we did that, we saw that there was clearly changes with these single amino acid changes. First, the mutations, both for muscular dystrophy and lipodystrophy, caused a lot more attachment sites. So normal lamin causes about 11,000 attachment sites between chromatin and the nuclear envelope, whereas these mutations double the number, suggesting that there's much more heterochromatin and a perturbation of how the chromatin interacts with the nuclear envelope during regeneration. So not the right places in DNA are subjected to epigenomics at the right time. We could then sequence all those and see all in the genome, which was the actual genes that were changing their interaction with the nuclear envelope. And what we found is that we could map those to developmental pathways. So the SOX and OCT4 pathway is a key one for many cell types to determine whether a cell that's undifferentiated becomes muscle or becomes fat or becomes heart through development. So we were seeing perturbations in these developmental pathways in muscle cells depending on the mutation. So then when we went back to our biopsy resource, so we had all the biopsies in our neuromuscular biopsy bank, and we could take patients that we knew their mutations, and we took patients with both the X-linked form with Emerin, the dominant form with Lamin, and then an unrelated dystrophy called FKRP versus controls. And we could confirm that in these biopsies, only the patients with nuclear envelope disorders show SOX2 developmental abnormalities. So confirming our model using patient tissues. So this is, I just wanted to use as an example, is that neuromuscular tissue bank became critical to validate a molecular model done in vitro. And so in conclusion, nuclear envelope laminopathies, are disorders of epigenomics and cell fate. Patients with specific mutations in nuclear envelope components acquire novel pathological cell fate, so the cells are going down the wrong epigenomics pathway due to inadequate nuclear envelope chromatin associations, provides an explanation for clinical variation. Different missense mutations interact with different lineage-specific networks in chromatin during development. And without neuromuscular banking, we would not be able to validate this molecular model. So in the remainder of the talk, I'll talk about drug development, gene therapy, and commercialization, and I'll talk about two different drugs, one that was recently approved and one that hopefully will be approved soon. And one talks about precision mutation targeted therapy, which is vitilarsen, and one is where we increase the understanding of how a well-known drug, corticosteroids, works in terms of different activities of corticosteroids, and then we're able to engineer a better corticosteroid that removed bone morbidities, and that's called vamorolone. And in each of these, we use different business models that use what is typically used. So I'll explain that briefly. So how exon skipping works, this precision targeting, is that the normal, this is to treat Duchenne muscular dystrophy. It's one of the most common genetic disorders worldwide and the most common form of muscular dystrophy. It's caused by the dystrophin gene or DMD gene. It's the largest gene in the human genome. And the gene has a large number of exons, 79 exons. And all those exons must be spliced together in muscle to create the messenger RNA. And here I've just diagrammed a few of the exons of the 79. And when they fit together to maintain the reading frame of the 1, 2, 3, 1, 2, 3, 1, 2, 3 amino acids to make the protein. So canonical DNA to RNA to protein. And that makes the dystrophin protein. And the dystrophin protein then associates with the plasma membrane and stabilizes the plasma membrane in muscle cells. In Duchenne muscular dystrophy patients, patients are missing one or more exons. And the remaining exons in the patient, when spliced together, are no longer in frame. So they do not fit together correctly. That means there's a frame shift. You cannot make protein from that RNA. So you have a loss of function. So Duchenne is then the loss of dystrophin and unstable plasma membranes. With these targeted therapies, our goal is to use systemic delivery of oligonucleotides that then go into the nucleus. The oligonucleotide drug binds an exon neighboring the deletion and blocks that inclusion of that exon in the RNA. So this forces the cell to skip exon 51 and instead splice 49 to 52, which are then in frame. So since you've restored the reading frame, you now can make the protein to the end, just missing some amino acids in the center. So this protein is semi-functional and stabilizes the plasma membrane. So I'll just briefly describe some recent studies of the first successful exon skipping drug in restoring relatively high levels of dystrophin, which is vitilarsen. So the first trial was done as 2 parallel trials in the US and Japan. So we ran the US one in the Cooperative International Neuromuscular Research Group, and our collaborator Shinichi Takeda in the National Center for Neuroscience and Psychiatry in Kodaira, Japan, ran the Japanese one, and these was in collaboration with NS Pharma, Nippon Shinyaku Pharma. So this was morpholino oligonucleotides against exon 53, systemic IV delivery of high doses weekly. And the primary outcome was a surrogate biomarker. So in the US and Japan, it's possible to get drug approval just showing you're bringing the missing protein back. So dystrophin replacement, with the anticipation that further trials will show clinical improvement later. So basically you get drug approval just on a biomarker, not on clinical improvement. So it's called accelerated approval. You then must do placebo-controlled studies later, post-marketing, to show clinical improvement. So we had the primary outcome of dystrophin increase in muscle, which was a surrogate outcome, and exploratory clinical outcomes of motor function tests. So after 6 months of treatment, the pre-treatment biopsies, of course, had no dystrophin because that defines Duchenne muscular dystrophy. But after treatment, we clearly see we're bringing dystrophin back to these patient muscles. And that was in all groups. You can also look at the RNA to see if the drug is hitting its target and inducing exon skipping. So that's by RT-PCR. And here you see before treatment, only the endogenous unskipped outer frame RNA. But after treatment, you see this new band, a new RNA missing exon 53. Again, direct evidence of the drug hitting its target. If we look then at 6 months after treatment, now this is not a placebo-controlled trial, but we used an external comparator, which is a Duchenne natural history study we ran within the Cooperative International Neuromuscular Research Group. 400 patients followed about 8 years internationally. And we see that the natural history patients over 6 months decline or barely stable, whereas the drug-treated patients clearly improve. Again, not double-blind, but suggesting that there is clinical improvement. And lastly, we just recently published 2-year follow-up in this initial cohort of patients. Again, external comparator. But again, we see that the vitilarsen patients improved and were stable, whereas the natural history patients declined with time. So in summary, this drug is safe. It's effective in drug-induced dystrophin replacement, 6% mean normal levels, suggestion of clinical efficacy in small trial versus external comparators, double-blind placebo-controlled trial is underway, and marketed by NS Pharma via accelerated development pathway, both in the US and Japan. And there was also an incentive from the Food and Drug Administration in the US, where they were awarded a rare pediatric disease orphan voucher, which is worth about $100 million that could be sold to another drug program. The last few slides, I'll talk about a second drug development program, which is vamorolone, which is a safer alternative to corticosteroids, and briefly summarize data from these 4 publications. So to remind you what corticosteroids look like, prednisone, prednisolone are among the most prescribed drugs worldwide. The corticosteroid anti-inflammatories, fastest Nobel Prize awarded back in 1950 for just a dozen arthritis patients that got much better with corticosteroids very quickly. And corticosteroids have both prodrug and active drug. And it all depends on this one oxygen here, which is the 11 beta position of the steroid. And if it's a hydroxyl group, it's active. If it's a carbonyl group, it's inactive and a prodrug. And this is true for all the types of corticosteroids. That conversion is mediated by these 2 enzymes throughout the body called the hydroxysteroid dehydrogenases. And there's at this 11 beta site, And one does the forward reaction from carbonyl to hydroxyl, and the second enzyme does the reverse reaction. So these are very important site here. And what we did was query if we take off that site. So vamorolone, the only change from vamorolone to prednisone is we remove that oxygen. We still bind the receptor, but we bind with slightly lower affinity. And so what I'll show is running this through the drug development pathway. So the first you can do is in vitro methods. And first, is it anti-inflammatory? And so the key anti-inflammatory activity is an inhibition of innate immunity through NF-kappa B. And in this NF-kappa B reporter assay, we see that prednisone and vamorolone and BBP15 are the same thing, are very similar. So we've retained that anti-inflammatory activity. Now the question is, have we differentiated in terms of safety? Our goal is to see if we can remove the safety problems such as bone morbidities, weak bones, breaking of bones. And so one activity we looked at was activation of gene transcription throughout the genome. And we see vamorolone has lost much of that gene transcription compared to classic corticosteroids. So we're seeing some differentiation of vamorolone from corticosteroids. There's two other activities we looked at. One was this HSD enzyme pathway I described earlier. And without this, we are no longer a substrate for these two enzymes. And lastly, we looked at the mineralocorticoid receptor. So corticosteroids often are agonists of the mineralocorticoid receptor that controls fluid balance and salt balance in the body, whereas vamorolone has an opposite effect. It is a potent antagonist of the mineralocorticoid receptor. So let's first study mice. We took mice with Duchenne muscular dystrophy, and we see we improve their strength, as does prednisone. And if we look at safety, one of the key safety concerns in children as corticosteroids is stunting of growth. So if you give children prednisone daily at a high dose, as they do with Duchenne dystrophy as standard of care, you stop growth, and children become quite growth stunted. So we see that even in mice with prednisone, after treatment, they're much smaller. But with vamorolone, also called BBP15, there's no growth stunting. So let's go to human trials. So we studied a broad dose range from 0.25 to 6 mg per kg of vamorolone. And here we're showing 6-minute walk tests, so how far a child can walk in 6 minutes. And we see clear dose-related improvements with time, over time with treatment. So this suggested that we were seeing benefit or efficacy. So we then just recently published a double-blind placebo-controlled trial, which was also had a prednisone arm. And what we see is that placebos show this deterioration over 6 months, where the drug treated clearly shows benefit by all outcome measures. So vamorolone met primary and 4 secondary outcomes. The 6 mg per kg, significant improvement versus placebo across all, and very similar to the prednisone group. But what about safety? That's our goal, is to improve safety. So what we found is we have lost bone morbidities. So bone biomarkers show no dealer changes with vamorolone, no stunting of growth in children, less vertebral fractures. So we have clearly differentiated vamorolone from corticosteroids. And my last slide is just to mention the drug development business models for these. The typical business model is academic groups do early preclinical stage, venture capital groups provide funding to move to early clinical stage, pharma purchases the asset, moves to late clinical stage and approval sales, but the business model all hinges on maximizing return on investment or making money. So with Vidal-Larsen, it was a close collaboration between Japan and US academics, use of academic networks for the clinical studies, no venture capital, and we were had more flexibility with robust trial design and dystrophin findings. And so the FDA said this was a robust study. They were well-designed trials, very clear dystrophin data, which was in contrast to other exon skipping studies. In the court of vamorolone, we took that one step further. The clinical program was retained by academics through late clinical development, no venture capital. And because of that, we did things that were very scientifically robust. And again, the findings, as you can see, were very clear. I'd just like to thank in the vamorolone program, the physician collaborators, neurologists throughout the world, and Michaela Guglieri at Newcastle and Paula Clemens at Pittsburgh that were the study chairs, and also the many foundations, nonprofit foundations and governments, including the European Commission, the National Institutes of Health, that made these programs possible. And lastly, for Vida Larson, Shinichi Takeda, my long-term friend and collaborator in Japan, Paula Clemens, and the Cooperative International Muscle Research Group and MS Forum. Thank you.
Thank you very much, Professor Hoffman, for the excellent presentation. If you have any questions from the audience, we can direct them to Professor Hoffman.
Thank you, sir, for this excellent presentation. So I'm Nalaka, one of the PhD students of Professor Rani. Let me ask one question on your recent drug, Vemurafenib. When I come across your recent article on Vemurafenib you published in JAMA, so I saw that there are very good results there. So recent literature says that inflammatory molecules that develop in peripheral will cross the blood-brain barrier and it will go to the brain of the muscular dystrophy patients. So it will increase the inflammation in brain cells and it will make the cognitive deficits in muscular dystrophy. So when I see the literature, I saw that the effect of Vemurafenib on brain inflammation is an untouched area. So if you can remember our recent publication, which you are also a co-author, so we identified a model with astrocytes, so that linked Alzheimer's disease and Duchenne muscular dystrophy in a cognitive model, and we identified some biomarkers that are related to cognition and inflammation. So my question is, is there any possibility to identify or further investigate the effect of this drug on the brain inflammation and cognition of muscular dystrophy? So this will not, if the results are good, it will not treat only the muscular dystrophy, but it will be a treatment for aging disorders. That is my question, sir.
No, that's an excellent point, and I certainly agree that that needs to be studied further. There is some suggestion both that prednisone may worsen intellectual function in Duchenne. That's a recent publication. And in vamorolone, we seem to show less mood disturbance in patients. So that holds promise. Also, the prednisone-mediated neuronal problems is thought to be mediated by that same 11-beta hydroxysteroid dehydrogenases, which we are not a substrate for. So I agree with you, there's certainly promise for testing vamorolone in CNS disorders. So it would be great to collaborate with you and others on that.
Thank you, sir, because I think we can do some further studies using our recently identified proteins. Thank you.
We have two questions from the audience.
I'm running this Silva. Can you hear me?
We can hear. President, please.
Yeah. Actually, Professor Hoffman, thank you very much for that very beautiful, simplified talk. What I would like to ask you is, you had just started, you isolated the disease-causing protein to a group of people who never had died. And there was no cure, no treatment, but then to translational.
I think you're breaking up. I can't hear him. Can others?
Can you hear me?
You broke up there for a while, so we couldn't hear the last.
So what I want to say is, from a protein of the disease you isolated, and then you came to translation way of developing gene therapy and a drug development, and how, from a Sri Lankan point of view, I mean, I have set up a brain bank and a DNA bank, and that's your support. We got from NIH long time ago, and we have around about 623 or 600 rare diseases, and of which 200 are eligible for gene therapy, and also we have for DMD as well. So how best we could link Sri Lanka and also the KDU and, Biobank with you in order to link form of clinical trials or gene therapy, so that we also could get involved in this because I mean, it's hard work that I've done and set up this entire national asset.
Yeah, I think the options are, one, to just do one-on-one collaborations with ourselves or others, and come up with specific experiments like we just talked about with Nalaka on, for example, brain studies. The other option is to make your resource open to a broad variety number of scientists worldwide. And so, like the French Muscular Dystrophy Association and Italy have been pioneers in developing supported resources to facilitate research worldwide. And I think I'd be delighted to help you find resources to provide like small portions of samples to other investigators, and the shipping and all that would need support, but I'd be happy to help you find support for that.
Okay, thank you very much.
Thank you very much. That will be great, and we in Sri Lanka will be able to, I mean, then move forward in this high-tech and clinical trials, and especially from a point of view of KDU.
Yes, so thank you very much, Professor Hoffman again. That brings us to the end of this section. So to sum up today's speeches, we had two presentations on nanomaterials delivered by Professor Nalin and Professor Lyudmila Turyanska. Both were talking about nanomaterials and their applications in drug delivery systems and as electronic devices and in tissue engineering and in medicine. So both were very inspiring speeches. And also it was highlighted the fact in Sri Lanka how there is a big gap between the scientists, the work that we are doing as academics, and how it is practically translated into the practical outcome. So how there is a big gap between the inventions and the commercialization. And also finally we listened to Professor Eric Hoffman's speech about the molecular based drug delivery systems for muscular dystrophy and neuromuscular disorders. And they were talking about the development of molecular based drugs which can be used as alternatives for currently available drugs, and also talking about the clinical trials and how it can be commercialized. So that had gone to a very later stage of the invention, which is very close to the drug being commercialized. Professor Nimal will sum up the other two.
Let me summarize the presentation by Dr. Andrés. So he has taken us through a very important area of the molecular medicine, namely gene therapy. It is becoming forefront of the treatment strategies for many genetic diseases, especially in the area of inborn errors of metabolism and many other genetic diseases. Even they are now going for research on diabetic mellitus type 1 and many other important pediatric diseases as well as adulthood diseases. In addition, he also highlighted some metabolism of liver metabolic diseases and bilirubin toxicity, neurotoxicity. You know, if the bilirubin is not broken down, there will be severe brain issues are coming. So it's very important area. So we thank him for his excellent presentation. And next come to Professor Sagarika Ekanayaka's presentation. So that is an eye-opener for all of you. Maybe some people are first time hearing this information about this parboiled. The word is very technical, so we can use for Sri Lankan slang, Nadu. So that is the best word that everybody, when you go to the any grocery or supermarket, you can see the in big letters, Nadu, various trade names. So go to that one and take that back, not to the other areas of Kiri Samba, and with this very, very affluent names. People go for Samba, otherwise Nadu, they don't care. But Nadu is the best for our health. I learned from Professor Sagarika Ekanayaka long time back. So we eat only Nadu. So it has become very good. You have seen the mechanism behind it. She has very nicely explained. The slow release of sugar not available to release immediately. So your blood will not be concentrated with glucose, and dietary fiber delays everything. So during the parboiling mechanism, everything comes up. So very important discussion. And even she has touched the traditional rice varieties. That is also now coming up from a long time, but the prices are exorbitant, very expensive still because of the short availability, high demand. So maybe 500, 600 at the current moment per kilo. So even traditional rice, you have to parboil in some cases. Okay, right. Okay, thank you very much. So this is the summing up of the, so everything is very timely topics for all of you. So especially the postgraduate students, you can catch up some ideas from these enlightened speakers. You can continue even collaborate with them. You can contact them, because now in a just Google search, you can find out the email address, and you can write to them their ideas, their papers, discussions.
Ladies and gentlemen, we have come to the end of the plenary session. Now it's time to appreciate our plenary speakers and the session chairs for their contribution towards the basic and applied sciences session of this conference. It was indeed an insightful and enlightening session. Next, I would like to respectfully invite our co-chairperson, Professor Nimal Punyasiri, to present the token of appreciation to Senior Professor Sagarika Ekanayaka. Senior Professor K.M. Nalin de Silva had left the session early. So I would like to call upon Professor Nimal Punyasiri to award the token of appreciation to Senior Professor Sagarika Ekanayaka. Thank you very much, sir. I would like to take this opportunity also to thank all the other plenary speakers for their wonderful insight and enlightenment towards the session. Senior Professor K.M. Nalin de Silva, Dr. Andrés Fernando Muro, Professor Lyudmila Turyanska, Senior Professor Sagarika Ekanayaka, and finally Professor Eric Hoffman. Now to award the co-chairpersons with a token of appreciation, I would like to cordially call upon the Assistant Director, KDU Care. I would like to call upon Professor Charitha L. Goonasekara and Professor Nimal Punyasiri to receive the awards. Thank you, sir and madam. We will now go for a short break, and after the break, we will be starting the poster presentation session. The technical sessions of the basic and applied sciences track will be held tomorrow, starting from 8:30 a.m. onwards. I invite all of you to participate for this session tomorrow, both in person and via online. The Zoom links for the tomorrow's technical session will be emailed to you today. Now I would like to invite you all for refreshments which are arranged outside, and after the break, we will be joining back with the poster presentation sessions. Thank you.
Good evening, ladies and gentlemen. We will now commence the poster presentation session of Basic and Applied Science track. We have three eminent panelists to lead the judging panel for the poster presentation session today, including Dr. Nadisha Ratuvadu, Dr. Peshara Kumari, and Dr. Amila Besekara. To begin the poster presentation today, we have our first poster containing the ID number 6 with the poster title, Presence of Collagen X in the Dentine of Developing Tooth, presented by K. Kaluarachi. The questions will not be asked as we are running out of time. In case, if any questions, the judging panel will be informed. K. Kaluarachi, we warmly invite you to present your poster.
Sorry. There are about 28 collagens in the body. Out of that, I am concerned about collagen type 10. Now, collagen type 10 is the major extracellular component of the hypertrophic chondrocytes, which are destined to be calcified. Usually, collagen 10 is found in the areas where they are going to be biomineralization. Now, in our study, we thought that in the body, when the cartilage is being calcified, collagen 10 is there. When other biomineral processes are taking place, collagen 10 is there. So what about tooth? Therefore, we took 18.5 days DPC mouse embryos, mouse embryo heads, and did some histology and carried out immunohistochemistry for collagen type 10. Now let me tell you that there are several teams in this connective tissue field have tried this before. They were failed. And I don't know why, but we were really successful. And if you see the figure 1, you see on your left-hand side, the calcified areas of the tooth germ of the 18.5 day PC mouse embryo. On the right-hand side, you see the signal for collagen type 10. The internal control would be the Meckel's cartilage present next to the tooth follicle. And in the higher magnification, in the figure 1, the lower panels, you see collagen type 10 is present in the odontoblast of the tooth germ. And if you see figure 2, if you look very carefully, the odontoblasts have the signal for collagen type 10, whereas the amyloblasts, the cells which are producing the enamel, they don't have the signal for collagen 10. Because I am mentioning this because previously one group has claimed that they have found collagen 10 in the amyloblast. And inside the pulp chamber also, you will see in the figure 2 lower panels, in the higher magnification, there are some signal for collagen type 10. Yeah, so that's my poster. I'm ready to take questions if you want.
Next, the poster ID 36, Screening of Phytochemical and Biomedical Effects of some Selected Psidium Species Leaf Ethanol Extraction by author M.S.K. Ilma.
Good evening, everyone. Actually, there's a Psidium species. There are the interest in Psidium species leaves using in the medical field. It has increased the interest, and in this study I have used three species of Psidium. Actually, Psidium genus, Psidium guajava, Psidium cinerum, the species we have used in this study. In those three species, actually, you can see the results. We have evaluated the ethanol extraction to manipulate in the antioxidant to get the antioxidant activity and anti-alpha amylase assay we have done, and then we have actually there's a result. It's like Psidium guinis show the somewhat similar, nearest results to the green tea. So I would like to suggest that Psidium guinis can be the TPC results. Actually, the 218.96 milligram that was somewhat similar to the green tea extract. So it can be used for the green tea in future. Actually, the Psidium leaves. In these three, Psidium guajava, it shows the anti-amylase assay was more compared to other two species. So compared to other two species, Psidium guajava can be used for the diabetics. With the further research, we need to do further research. With that, we can use this for to treat any treatments for diabetics, because the anti-alpha amylase assay 50% in which I see 50 results was high in this Psidium guajava. And Psidium guinis and Psidium guajava was have given the somewhat actually the antioxidant assays with high results compared to other Psidium cinerum. And that's the whole thing. Even in the TPs, phytochemicals and flavonoid contexts were high in Psidium guinis. You can see through this result.
And Cydium, actually, there's about like 150 species worldwide. In that, only like 20 species were using to the edible fruits. So, thank you.
Next, we have poster ID 38, review on wastewater treatment using constructed wetland systems, implications from Sri Lanka, presented by AMSN Amarkon.
Good evening, everyone. I'm Soumya Amarkon from Asian Institute of Technology. Here I'm going to review on the wastewater treatment using constructed wetland systems. Mainly I'm focusing on the implications from Sri Lanka. Actually, the constructed wetland system is somewhat popular in Sri Lanka, but not using in a effective manner. So here I have chosen some wastewaters like slaughterhouse wastewater and leachate, and also textile wastewater. So I have review on how they have used these constructed wetlands to treat these kind of wastewaters. So in that I have found that floating wetlands are usable in those kind of things in Sri Lanka, and they are using effectively floating wetlands here. And I have found some of the macrophytes that mainly used for the constructed wetland systems. And here I like to give a recommendation. The constructed wetland is a usable system for the wastewater treatment, but in Sri Lanka we need to develop that method in a effective manner. Thank you.
Next of all, we have poster ID 86 with the title Comparison of Phytochemicals and Antioxidant Activity of the Polysaccharide and Depolysaccharide Methanol Extracts of Brown Seaweed Nucifera minima, presented by H.S. Kumarasinghe.
In this study, it intended to develop, explore, compare, and contrast compare and contrast the phytochemical composition and antioxidant activities of polysaccharide and depolysaccharide samples and fractions of Sri Lankan marine alga, Ginospora minima, and to investigate the best sample out of them in order to examine their potent incorporation in pharmaceutical, cosmeceutical, and nutraceutical industry applications. So, for this study, we selected Sri Lankan Ginospora minima, as it founded as an ideal anti-cancer agent with potent antioxidant and phytochemical capacities. So previous studies and findings related to this alga revealed that its depolysaccharide portion composed with high phytochemical composition along with potent antioxidant capacity. So we needed to find whether what is the importance of polysaccharide portion with this antioxidant activity and how it affects for the composition of available phytochemicals. So we decided to compare and contrast the phytochemical composition and antioxidant activity of two samples, polysaccharide and depolysaccharide one, to find the best one for further studies. As you can see that this image shows how we follow the procedure to get these results, and so these are the results we obtained after following this process separately. So based on the results, it is found that the hexane fraction of both extracts is the least active fraction for all antioxidant assays. and ethyl acetate fractions of both extracts are the ideal fractions with potential antioxidant activities. So by looking at these results, we concluded that the depolysaccharide C-minima crude methanol extract and its fractions exhibited a high level of antioxidant activity along with high content of phytochemicals. So which we can develop further be utilized to determine the bioactivities that lead to future drug development. Thank you.
Next, we have poster ID 95 with the title In Vitro Bioactivity of Methanol Extracts of Elaeocarpus certus leaves and Fruit Bioactivity of Methanol Extracts of Elaeocarpus certus leaves and fruit, presented by A.I. Kurupu. A.I. Kurupu, we are unable to hear you. A.I. Kurupu, we are still unable to hear you.
Am I audible?
Yes, now we can hear you.
Okay, so good evening, everybody. My topic is in vitro bioactivity of methanol extracts of E. serratus leaves and fruit. E. serratus is known as Verelu in Sinhalese, and this plant is an underutilized or neglected plant in Sri Lanka. And it's known to have many medicinal properties and a fair amount of fiber and oil and etc. However, this plant is not widely known, and it's actually not found commercially. Not just Verelu, there are about 60 such plants in Sri Lanka. So we selected Verelu and determined the bioactivity of it. So initially what we did was we selected leaves and fruit, and it was subjected to methanol extractions. We then determined the total polyphenolic content and the flavonoid content, and we found that the fruit was actually better than the leaf because it had higher amount of polyphenols. We then went on looking at the antioxidant activity, and very interestingly, we found that the fruit actually has a very high antioxidant content, which was about 5 times more compared to the leaf, using the DPPH assay. We then looked at the ORAC assay, which is also an antioxidant assay. And here actually we found that the leaf had more activity than the fruit, but anyway it was kind of low. So these two assays are actually, both assays are antioxidant assays, but they are actually different. So ORAC measures the polyphenol's ability to transfer hydrogen atoms to oxygen radicals, while DPPH measures the electron transfer reaction between DPPH radicals and the polyphenols. So then we moved on looking at the mineral content of the leaf extract. And interestingly, again, we found that it had a very high sodium content and some other minerals as well. We also looked at the anti-inflammatory activity using two bioassays, which was the human red blood cell membrane stabilization assay and the egg albumin assay, using both the leaf and the fruit extracts. And we found that the fruit was much more better than the leaf, but both of them were quite good. But we found that the fruit had more anti-inflammatory activity in both assays compared to the leaf. We also looked at the toxicity of these extracts using mammalian cells. We used Vero cells, and we found slight moderate, probably moderate toxicity. So we did this because we wanted to actually finally go into a food supplement or make actually value-added products. So we need to find whether it has any toxicity. So these results actually suggest that we can actually make maybe using the fruits, maybe we can make a pickle or a jam, which is a value-added product, and it actually might help the Sri Lankan economic revenue. So this is actually a basic study, but we hope to do more studies with this underutilized plant.
Next, we have poster ID 97 with the title Microclimatic Buffering Potential of Forest Type in Wasgamua National Park, Sri Lanka, presented by A.D. Senaviratna.
So good afternoon, everyone. My research is about the microclimatic buffering potential of forest types in the Wasgamua National Park, Sri Lanka. The objective of this study was to document the variation of the microclimatic buffering potential of different habitats within the dry zone landscape. So in this study, I conducted this study at the end of the September and end of the March. That means beginning of the dry season and end of the dry season. Then 45 sampling sites were randomly picked by maintaining a minimum distance of 2 kilometers between each. And then I recorded temperature and relative humidity values by using sensors that recorded data at every 30 minutes for a period of 24 hours. Then I calculated vapor pressure deficit values by using this equation for all habitats. That means for grassland, for scrub forest, dry mix evergreen forest, and riverine forest. So the highest VPT values were recorded at the grassland for the both habitat, for the both levels, and the lowest was recorded in the riverine forest. And according to the graph, if the line for the two habitats is placed below the one-to-one line, that means that habitat got moisture conditions than the reference site. And if the line was placed above the one-to-one line, that means that habitat got drier conditions than the reference site. So in this study, the lines for all habitats were placed below the one-to-one line, indicating that they have moist conditions than the grassland. So, and from the area between two lines, we can calculate the microclimatic buffering potential. So greater the area, higher the microclimatic buffering potential, and vice versa. So in here, riverine forest got the highest buffering capacity value, where the scrub forest got the lowest. So this study provides evidence in support of the hypothesis that there are considerable differences in the microclimatic buffering capacities among the different habitat types. Thank you.
Next of all, we have poster ID 178 with the title Biodegradation of Burnt Engine Oil by Surfactant Producing Marine Isolates of Pseudomonas species, presented by J. Matushika.
Good evening, everyone. I'm Judy Madhushika, a MPhil student working at KDU Care. And this preliminary study is on biodegradation of burnt engine oil by surfactant-producing marine isolates of Pseudomonas species. As we all know, burnt engine oil pollution in marine environment has now become one of the most threatening environmental issues. So, bioremediation by microbial degradation can be considered as one of the most environmental friendly approaches to tackle this issue. So basically in this study, we aim to assess the potential of our newly isolated Pseudomonas species in degrading the burned engine oil under fed batch experiments, and also to determine their capacity to produce biosurfactants during the biodegradation. So basically for this study, a 1 liter fed batch reactor was made with 450 milliliter of minimal salt media, and 2% of burned engine oil was added to be used as sources of carbon and energy by the microbes, and also 2% of microbial inoculum of Pseudomonas species was added, and the reactor was incubated up to 30 days at ambient temperature with continuous stirring to show and to get an idea of the microbial growth pattern during the incubation. Viable cell colony counts were recorded on the 10th, 20th, and 30th days of incubation. And also preliminary biosurfactant assays, which are drop collapse method, oil spreading assay, and emulsification assays were performed to check the presence of biosurfactants. For that, 1% of SDS and distilled water were used as the positive and negative controls respectively. So moving on to the results, our Pseudomonas species was able to utilize the burnt engine oil as sole source of carbon and energy. This was indicated by the high viable cell counts during our fed batch experiments. And the preliminary biosurfactant assays results were also confirmed the presence of biosurfactant. In this picture, we can see the results of the drop collapse method, which is the first set. There we can see A and C, which are our positive control and sample, respectively, got collapsed on the oil surface, whereas the negative control did not get collapsed. And the second set pictures are for the oil spreading assay. There also we can see D and F, our sample and the positive control displaced the oil and made hollows, whereas the negative control did not do so. These results confirm the presence of biosurfactants, and from these we can say our Pseudomonas species can be suggested as a good candidate to develop a cost-effective method for the bioremediation of burnt engine oil. And currently we are doing further studies to analyze the burnt engine oil, degradation profiles of the burnt engine oil, and also proceeding this research further with the characterization of biosurfactant will be done. Thank you.
Next, post ID 180 with the post title Formulation and Sensory Evaluation of Malted Drink Powder incorporated with selected legumes, seeds and rhizomes, presented by MWP Sandupama.
Permission from everyone. My post presentation is on formulation and sensory evaluation of a malted drink powder incorporated with selected legumes, seeds, and rhizomes. As you know, there's a wide range of malted drink products available in the market, and most of them are added with palm oil as their major fat source. So the regular consumption may negatively affect the cardiovascular health of consumers. Therefore, the formulation of a malted drink with soybean flour, malted green gram flour, ginger powder, and watermelon seed flour as a substitute for palm oil is identified as a worthwhile focus. Research methodology. Taguchi's L8 orthogonal array was selected as the best experimental setting for this product development process. For that, we have to determine high and low levels of each variable, and it was done by performing trial and error experiments. Then by substituting those values, the design matrix for the product formulation was accomplished. As the next phase, sensory evaluation of those developed 8 formulations was done. It was done under 5-point hedonic scale, and 5 major attributes, namely appearance, aroma, taste, mouthfeel, and overall acceptability, were assessed by a semi-trained panel of 30 members. Sensory evaluation was held in 3 sessions since there were 8 formulations. In the first 2 sessions, the 8 formulations were evaluated 4 by 4. Then the best 2 formulations screened from those 2 sessions were evaluated in the final session, and the best formula among all were selected by statistically analyzing the sensory data. Results and discussion, the ingredient ratios for the 8 molded drink formulas were obtained as indicated in the table 1. As the results of first 2 sensory sessions, it was determined that the formulation 3 is the best out of first 4 formulas, and formulation 7 was the best among last 4 formulas in terms of all the sensory attributes. Then the selected 2 formulas were assessed in the last session, and as the final outcome, formulation 3 was selected as the best formula in terms of all the 5 sensory attributes. Figure 1 illustrates the 3 web diagrams developed for the 3 sensory sessions, and they clearly depicts that the best formula in each session. So as the conclusion, formulation 3, which has 52.17% of soybean flour, 26% of molded green gram flour, 17.39% of watermelon seed flour, and 4.34% of ginger powder was selected as the most sensorially accepted formulation. Thank you.
Next of all, we have poster ID 230 with the poster title Determination of Response of Selected Rice Genotypes of Planting Date During Maha Season on Selected Agronomic and Yield Characters under Greenhouse Condition, presented by HMASP Jayasinghe.
Right. I am Arun Jayasinghe from Faculty of Agriculture, University of Rona. Rice cultivation is a major livelihood of people in Kamrupeti area, and they mainly rely on new input rice cultivation. However, their fields are frequently affected by biotic and abiotic stresses, for example, flooding and BPH attack. According to literature, Mavi is a set of traditional rice land races which are tolerant to flooding and BPH attack. So this experiment was conducted to determine the effect of planting date or natural photoperiod variation, because Mavi is a photoperiod sensitive variety with short day sensitivity. Exposure to long days prevent or delay flowering. 2 Mahamavi accessions and 1 Mavi Samba accession were selected, and BG300 was selected as the check variety. 4 planting dates were selected from mid of the September to mid of the November, with 2 weeks interval between 2 planting dates. In each planting date, 2 weeks old seedlings were transplanted in pots following complete randomized design with 5 replicates. And as growth parameters, days to 5th leaf, height at 5th leaf, and days to flowering, plant height at flowering. And as yield parameters, days to grains per panicle and effective tillers at harvest was evaluated. And the results was the lowest growth parameters was from the first planting date for all tested growth parameters, and the highest values for those yield parameters was growth parameters was from the 4th planting date. In contrast to that, highest yield parameters such as grains per panicle and number of effective layers at harvest was from the first planting date, while the lowest was from the fourth planting date. So I can conclude the findings. So the best planting date for the selected Mavi accessions is the first planting date, the mid of the September, and the findings can be verified by repeated trials in different places by pot experiment as well as the field trials, and there is a need of incorporating planting date for breeding programs of Mavi varieties. Thank you.
Next, poster ID 272 with that poster title, Evaluation of Sri Lanka Wild Fruits based on Free Radical Scavenging Activity, Polyphenolic Content and Cytotoxic Activity, presented by HNI Abesirivardhana.
Good evening, all. My topic is about evaluation of Sri Lankan wild fruits based on the free radical scavenging activity, phylogenetic content, and cytotoxicity activity. In this study, we focus to free radical chemistry that represent an emerging trend in nutraceutical studies, because numerous studies suggested that antioxidants can interact with the free radicals in a way that can prevent from damaging biological compounds. In diverse of variety, there are diverse of varieties, synthetic and natural antioxidants have been shown therapeutic impacts in various diseases, but the synthetic antioxidants are examined as a high toxic and having severe side effects. So as a Sri Lankan, we are proud nation of enriched over 120 species of wild fruits. They are great resources of polyphenolic and flavonoids and the high amount of secondary metabolites. So in this study, we aim to study and focus polyphenolic and the flavonoids and the antioxidant content in the nutraceutical properties in the Sri Lankan underutilized wild fruits, such as we selected three wild fruits in 3 different families. They are in singular names. They are Kahata fruit and the Kahata fruit and Hindang and the Atamba fruit. Hello. And in conclusion, we reveal that ethyl acetate and aqueous fractions of Kahata fruit have highest antioxidant.
HNI Abesirivardhana, hope you are done with your presentation.
Yeah.
We are unable to hear you, Abesirivardhana.
Hello.
Yes.
Can you hear me?
Yes, we can hear you. Hope you are done with your presentation.
Yes.
Yes, thank you. Next, we have poster ID 339, relationship between selected anthropometric parameters and 50-meter freestyle swimming time in teenage swimmers, presented by RMUS Sugatadasa.
Good evening all. I am RMUS Sugatadasa from Sabaragamu University of Sri Lanka. My research topic is relationship between 50-meter freestyle swimming time and the anthropometric parameters in teenage swimmers. Swimming is the most competitive event in the Olympic, and it consists with 4 strokes such as freestyle, backstroke, breaststroke, and the butterfly stroke. 50-meter freestyle swimming race is the fastest and shortest event at the Olympic, and there are so many factors affecting to the swimming performance time. The anthropometric parameters are the one of main factor that affect into the swimming performance, and anthropometric parameters are the non-invasive quantitative measurements of the body, including height, weight, length measurements, girth measurements, and skin fold measurements. Therefore, my main objective is of this study is to identify the relationship between selected anthropometric parameters and 50-meter freestyle swimming time in teenage male swimmers, and therefore I selected 30 teenage male swimmers as my sample. Then I tested their 50-meter freestyle swimming time and the 13 anthropometric parameter sides of their body. Then I got results from SPSS software, and the results indicated that their 50-meter freestyle swimming mean time is the 39.9 seconds, and there is a statistically significant relationship, moderate positive relationship between the 50-meter freestyle swimming time and the body weight, height, and the hand length. Most of the previous studies has reported that there are so many anthropometric parameters have a relationship between the 50-meter freestyle swimming time, and this study has concluded that there is a relationship has 50-meter freestyle swimming time and the body weight, height, and the hand length at the 0.05 level of significance. And these studies can be concluded that there are so many factors affecting to the swimming performance, and the teenage male swimmers have to manage their anthropometric parameter sites to achieve their targets, apart from the other influencing factors that affect into the swimming performance time. And I recommend this study to female teenage swimmers also. Thank you.
Next, poster ID 353 with the poster title, A Comparative Study on Phytochemical Screening, Antioxidant Activity, and Photoprotective Property of Ethanolic Extracts of Peel and Pulp of Mango, presented by SDAE Ranasinghe.
Good evening, everybody. My research topic was a comparative study on phytochemical screening, antioxidant activity, and photoprotective property of ethanolic extracts of peel and pulp of mango, Mangifera indica L cultivar Tommie-JC. Tommie-JC mango is a locally grown novel cultivar. Less research has been conducted to determine the photoprotective and nutraceutical activity of peel and pulp of Tommie-JC mango. So here we mainly focused to determine the antioxidant activity, photoprotective property, and to investigate the nutraceutical properties of peel of the Tommie-JC mango peel, and also to compare that qualities with that of the pulp. According to the methodology, first of all, the fresh ripe mango fruits were collected from different local markets in Gampaha district, and they were pre-treated. And then we took 20 grams of mango peel and 20 grams of mango pulp, and we separated them separately using a Soxhlet extraction using ethanol as the solvent, and for about 6 hours. And for the extract, we done phytochemical screening by standard procedures, and also we did total phenolic content and total flavonoid content using the Pollens Geocalcio assay and aluminum chloride colorimetric assay separately, and crude fiber content was determined using the AOSC method, and antioxidant activity was determined using the DBPH free radical scavenging assay, and also the SPF value was determined using spectrophotometrically. And also finally GCMS analysis was done only for the peel extract. According to the results, phenolics, proteins, and some other phytochemicals were present in both mango peel and pulp. And according to the TPC, TFC, and crude fiber results, the total phenolic content, the total flavonoid content, and crude fiber content was higher in the peel than that of the pulp. And according to the DPPH radical scavenging activity, when looking at the IC50 values, the IC50 value of the peel extract was lower than that of the pulp extract. So here we can determine that the peel extract having the higher antioxidant activity than that of the pulp. And also for the SPF value, we got a higher SPF value for the peel extract than that of the pulp extract. And as a conclusion, we can come to a conclusion that total phenolic content, total flavonoid content, crude fiber content is higher in peel extract, and it can be used as a food additive, and also can be used in herbal, nutraceutical, and cosmeceutical industries. Thank you very much.
Next, post ID 357 with the poster title, Forecasting of Female Labor Force Participation Rate, Data with Missing Values, Imputation, Sri Lanka, presented by N.N. Ranasinghe.
My study is female labor force participation rate with missing value, forecast in female labor force participation rate with missing value imputation. Female labor force participation rate is proportion of female labor force to total working age population. Here I used female labor force participation quarterly data which published by Department of Census and Statistics from 2004 to 2022. Here, but 8 data points were missing. Therefore, this analysis was done under 2 parts. The first part is missing value imputation, and the second part is fit-in ARIMA models. When I did this missing value imputation part, I used 9 imputation method in impute TS package in R software. Then the missing values were generated using Bernoulli distribution and under 4 missing rates and using 30 random seeds. Then data were divided into 2 parts as a training set and the test data set. Then the ARIMA model, the function auto ARIMA and the ARIMA was used to fit different order ARIMA models. In this result, you can see the MAPE plot which construct for the imputed missing values. Here, by considering error distribution, estimated the moving average method, estimated weighted moving average method was the best method to impute these results. Then in table 2, you can see the coefficient of fitted ARIMA model. Here I select this ARIMA model considering minimum AIC value and the significant coefficient. Then the table 2 represent the forecasted values of the female labor force participation rate for the next 6 quarters. Then I measure the error accuracy of fitted values using test data. My conclusion is we can use exponential weighted moving average method to impute missing values in this the female labor force participation rate data set, and we can use ARIMA 111 without drift model to forecast our female labor force data in study period, and the end of this year we can conclude 34.2% female labor force participation rate. Thank you.
Next, poster ID 377 with the poster title Physicochemical and Microbial Analysis of Morinda citrifolia Fruit Extract, presented by WMD Clinical.
Good evening to all of you. My research topic is physiochemical and microbial analysis of Morinda citrifolia, noni, and Indian mulberry fruit extract. So the main introduction is Morinda citrifolia is commonly known as Indian mulberry, and in Sri Lankan context it is known as ahu. So in Sri Lankan context it is lack of research publications, and also